Bienvenidos y bienvenidas al segundo día del coloquio de Quimioinformática e Inteligencia Artificial. Good morning or good afternoon, everyone. Welcome to the second day of the Coloquio of Quimioinformatics and Artificial Intelligence. Para continuar con nuestro programa, la maestra Fernanda Saldívar hará la presentación de los ponentes del primer bloque. Muchas gracias, maestra Fernanda.
Muchas gracias, doctor José Luis, por la presentación. Para mí es un gusto poder presentar a nuestro primer invitado del día de hoy, el doctor Miquel Durán Frigola. Estoy segura que a más de uno le va a interesar la investigación del doctor Miquel y la iniciativa de Ercilia, con la cual se están aplicando conocimientos de ciencias de datos al estudio de enfermedades desatendidas en entornos de poco financiamiento. Thank you for joining us of the second day of this Quimioinformática and Artificial Intelligence Colloquium. Our first speaker is Dr. Miquel Durán Frigola. Miquel's research interests lie at the intersection between drug discovery and large-scale biological data analysis. During his PhD studies and early career, Miquel developed several in silico methods for pharmacology, producing scientific publications in a broad range of topics, from theoretical chemistry to cell-based data analysis. Along this process, he has worked at IRB Barcelona, the Massachusetts Institute of Technology, the MIT, Tel Aviv University, IS Global ICSM in Mozambique, CIDIRC in Zambia, H3D in South Africa, and CMM in Austria. At IRB Barcelona, Miquel led the development of the Chemical Checker, a data-driven drug discovery tool that collects and harmonizes a massive amount of activity data, making it ready for machine learning tasks. Currently, Miquel is lead science at the Ercilia open source initiative. With Ercilia, he hopes to apply his data science skills in underfunded settings, such as research institutes in low and middle-income countries. Beyond science, Miquel has a master's degree in creative writing and studies humanities, and has published a novel and several short stories in Catalan and Spanish. Dr. Miquel, welcome, and thank you very much for joining us in this colloquium.
Many thanks, Fernanda, for this very kind introduction. Muchas gracias. So I'm going to share screen now. Let's see if it works. Okay, does it work for everyone?
Yes.
Can you see my screen and also my pointer?
Yes, we can.
Fantastic. Hi, everyone. It's an absolute honor to be here presenting and actually opening the second day of this colloquium. I'm Miquel. I will talk about Ercilia, which is a non-profit organization that we have recently launched. Based on the work that I did during my PhD and also postdoc times. Okay, so today you will learn a little bit about Ercilia, and also you will learn a little bit about my previous work as a postdoc, where I applied my knowledge in systems biology combined with chemoinformatics. Okay, of course you will also see a little bit of artificial intelligence and machine learning during the talk. So the outline of the talk today. Just in the beginning, I'm just going to explain briefly what the Ercilia is about. Then we will move towards the main asset that we are trying to develop for this nonprofit called the Ercilia Model Hub. Then I will introduce a few concepts for data-driven drug discovery that hopefully will help us produce decent or good quality models at scale. And finally, I will just mention some of the upcoming projects that we have for this incipient initiative, which is called Ercilia. So let's focus first on Ercilia. Ercilia is, as I said, a tech nonprofit. We started 2 years ago. Our mission is to strengthen, as Fernanda has very nicely explained, to strengthen the research capacity against infectious diseases and neglected diseases in low and middle income countries. And our vision is that of a world with more egalitarian access to healthcare. So if you look at the world as it is today, you can see and color countries by income. You can see that basically in the global north, we have the highest income, and in the global south, mostly in Africa, is where we find most of the countries with low to middle income countries. Okay. If we then focus, plot the wall not in proportion of the land area, but in proportion of the prevalence of infectious diseases, you can see we get a picture like this, where basically, as it's very apparent, mostly the countries that are most affected by infectious diseases are the African countries. That's very severe. And if we plot the world by the number of scientific publications, that is the scientific productivity, the amount of research that is being conducted, you see that Africa dramatically shrinks, and most of the research happens in the global north. This, of course, causes a severe imbalance in the type of research that we are doing and in the type of scientific questions and biomedical questions that we are asking. The goal of Ercilia is to tackle this inequity in the research ecosystem. Just to put some numbers, as of today, less than 5% of the research worldwide is produced in low-income countries. Only six of the 10 top causes of death in low-income countries are still infections. However, only 10% of the drugs that are currently under development are tackling infectious diseases. Okay, so these are a bit the numbers that we have for this inequity. At Ercilia, we are trying to tackle this problem precisely. How do we do it? Well, we do it computationally. This is why we are here today. And we focus on 3 pillars. The first is that we produce computational assets that are free and open source. We try to share our code as much as possible. We make the most permissive licenses possible. We don't aim at patents, and we, of course, aim at reproducibility of research. This is the only way we believe to make an impact in countries that are underserved and underfunded. Then, very important, the central pillar of Ercilia is that we produce research in country. We want to avoid this phenomenon called helicopter research, where basically the data is produced in the global south, and then this data is kind of travels to the global north, where it's analyzed and ultimately dies. We want the data to stay in the global south and to be exploited in the global south. This is why basically at Ercilia we are quite literally itinerant. We travel all the time and we perform our research in country. We want the science, or we hope that the science will be led by local institutes, not by institutes from the global south. And as I said, all the implementations, or most of them, we try to do them in situ, on premises. Finally, the third pillar is that we aim at sustainable collaborations. This means that whenever we go to a research institute in the global south, for instance, next week we are going to Cameroon, we provide a great deal of capacity building activities. We try to identify and train local champions, people that will eventually be able to adopt the tools that we might develop with them, and we focus our machine learning tools and pipelines to the settings where we are actually aiming at, which are typically low resource, right? So the machine learning that we do is typically quite light, as opposed to the GPU demanding machine learning that we are able to do in the global north. All right, so of course, this is a big mission. This is difficult to do, especially if you are just a small tech nonprofit that started a few 2 years ago. So of course, the first question that we had is, where do we start? What scientific problems do we tackle, or at least what area of biomedicine do we tackle? Well, we decided to start with machine learning. Why machine learning? Because machine learning, beyond the hype about machine learning, it has some advantages that can be quite important when we are talking about research institutes in the global south. It can be quite cost effective. Implementation of hypotheses and testing of hypotheses is relatively fast with machine learning. You can do it remotely when necessary. It's sort of domain agnostic, meaning that if we develop tools for one particular domain, perhaps we can translate them to other domains. And we believe there's a need for demystifying machine learning, and which is what we are doing here in this colloquium, right? Trying to make machine learning less cryptic and more understandable for everyone. On the biomedical side, we focused on drug discovery, mainly because I personally have a track record in drug discovery as a PhD researcher and also as a postdoc. We believe that drug discovery, and especially small molecule drug discovery, is still the go-to solution for many of the neglected diseases. Hopefully, we will be able to develop cheap medicines, and there are already some inspiring initiatives like medicines for malaria DNDi open source malaria that we believe we can build upon. The goal of machine learning applied to drug discovery, I think that can be nicely summarized in this graph, where we hope that compared to traditional drug discovery, we will reduce the time that it's going to take for discovering a new drug while being able to screen more molecules. Okay, so what are we doing in practice? What we are trying to do is what is called a model hub, a hub of pre-trained machine learning models that we will eventually use for our drug discovery tasks. Just to make sure that everyone is on the same page, I know I don't need to spend a lot of time in this slide. This is what we mean by a machine learning task. It's basically QSAR most of the times. We gather experimental information. For instance, here you can see several compounds screened against E. coli, and you can see that bacterial growth in purple. So if you expose E. coli to a certain compound and they still continue to grow, this compound can be called inactive. If the bacteria starts dying in a dose-dependent manner, then you can call this compound active, right? At the end of the day, we are transforming an experimental setup into an output. In this case, I binarized this output for simplicity, active or inactive, for each of the molecules. Okay, this is typically the entry point for any machine learning model that we want to build. What do we do after that? Well, we train a neural network typically. So the way we do it is the classical way of supervised machine learning. We have a training set. In this case, these 4 molecules. Some are active and some are inactive. First step, and the most fun one, I would say, and one of the most interesting one is featurizing the molecule. So featurizing means how do we represent the molecule in a format that the computer can understand, right? How do we go from a drawing or a 2D structure of the molecule to a numerical vector or embedding that the computer can use comfortably as an input for a machine learning model. Yeah, this first step, we do a lot of effort, as you will see, in getting this first step right. After we have a numerical representation of the molecule, then we just fit a model. It can be a neural network, can be a gradient boosting machine, can be anything really, right? So that we learn how to map from the numerical representation of the molecules to the output. The hope is, of course, that when we come with a new molecule, we will first feature the molecule, and then by looking at the vector of the molecule for which we don't know the activity, we will be able to make a prediction. Yeah. So this is what supervised machine learning is all about. And this is what we do at Ercilia 90% of the time. Now, what is special about Ercilia, or what makes this model hub unique? I think that what makes this model hub unique is this outer layer of the machine learning cycle. So the machine learning cycle, as you may know, almost always starts with raw data that almost always you have to process, that is clean and harmonized. Once you have processed data, typically you train a machine learning model that you have to tune and validate. Typically you do this with cross-validation. With your internal data set, you do a train set and a test set, and then you use this to kind of assess the performance of your model. Hopefully then you are able to do some external validation, and hopefully you are even able to do some experiments that will validate your model prospectively. Most of the research that happens in academia ends here. Yeah, ends with this validation and a nice scientific publication, hopefully. Many times the code is released in GitHub, and many times the data are released in GitHub, but what's not released most of the times is a tool that someone who is not a computer science can run, right? So this is this outer layer, which we call deployment. And by deployment, it means making the model ready for someone who is not an expert to run it without big efforts. This is critical in the global south, and is even more critical in the global south. In the global north is critical, but it's even more critical in the global south, where most of the people do not have data science skills. At the end of the day, what you want the experimentalist to do is to have an input, a molecule, for instance, and to get an output without doing much more. Okay, in this case, the predicted targets of this molecule. So how does it happen in practice? Well, we have different types of models in our hub. sometimes we just inherit models from the scientific literature. You might know this paper, Cell paper, very nice paper from 2020 for antimicrobial drug discovery. They produced a very nice neural network and a model called ChemProt, and they trained it on a data set of E. coli, like the one I was presenting, and they released the code and the pre-trained parameters, right? So at Cilia, the only thing we have to do is to provide an interface where the researcher can input their molecule of interest and they will get a prediction, okay? Sometimes life is not so easy. Sometimes we don't have models pre-trained and we have to train the models ourselves. For instance, in this case, this is a 2018 paper published in Science, doing a tour de force in screening a large collection of molecules. I think it was 500,000 compounds for their antimalarial activity. So very nice data set. However, if your molecule is not in that data set, this data set is perhaps not too useful. So we built a machine learning model on top of this data set so that when a researcher comes with a new molecule that was not screened in this paper, they will still be able to get a prediction based on the machine learning model that we've trained, okay? This is what we call in-house machine learning, where we build the machine learning model and we fetch the data from somewhere else. And many times, of course, what we're actually doing is doing machine learning in collaboration. In this case, we partner with someone, for instance, University of Buea in Cameroon. They want to predict, in this case, natural product activity, as you will see later on. So we build the machine learning model for them so that they can input a compound and they can get a prediction for whatever activity they are aiming at, okay? All right, so this is the Cilia Model Hub, and this is a mock-up of how it looks at the moment. As you can see here, there are several cards. Each card is one model. Yeah. For instance, there's a model related to predicting toxicity and synthetic accessibility of compounds. There's a model for log-P. There's a model even for compound price prediction. There's a model for antimicrobial activity, etc., etc., etc. Our aim is to do this as a mission, philanthropically, to screen the scientific literature and incorporate as many models as we can from the literature. We estimate that by the end of the year, we will have between 300 and 500 models already available in the Model Hub. Okay, so how do we build the in-house models and how do we build the machine learning models in collaboration, right? It's nice when we can inherit models from the literature, but most of the times we need to build the models ourselves because there's nothing available yet. So how do we make sure that we can build models that are quote-unquote state-of-the-art and quote-unquote scalable, meaning that we can build many models with the limited infrastructure that we have. And here I have to switch gears and I have to talk about this concept of knowledge representation, which before I was calling it featureization, right? So this is I think the key step of the Cilia technology. How do we represent the small molecules so that the modeling step becomes as easy as possible? So when we talk about knowledge representation, basically what we mean here in this context is how to represent numerically a small molecule. And traditionally, what we have been doing when it comes to knowledge representation, it's structural biology, right? So, or even chemistry, right? So basically, very explicit representations of compounds, in this case, which are very easy to understand even visually, right? In this case, we have a target and we have a compound and we can see very clearly where the compound binds and how it binds. This is great, and this is actually a very compelling way of representing knowledge. However, when it comes to machine learning tasks, as you know, knowledge representation typically becomes much more abstract, much more difficult to understand, because we deal with this concept of embeddings, we deal with this concept of tensors, etc. Right? So knowledge representation when it comes to machine learning is more abstract, and we need to make ourselves comfortable with it. Let me go a little bit more in detail. How have we been doing knowledge representation for years in chemoinformatics? Well, typically, I'm sure you're familiar with this concept, is through chemical fingerprints, or chemical descriptors, right? What is a fingerprint? What is a descriptor? There's many flavors of it, but at the end of the day, what you do is you have a chemical structure and you try to identify features, functional groups, traits, properties in this chemical structure that you map into a vector. Yeah. In this case, I'm just suggesting a fingerprint where if I find a particular pair of atoms, I just map it in the position correspondingly. Yeah. So because there is a chloride bound to an aromatic carbon, then I can put a 1 here. Because there is a nitrogen here, I can put the tertiary nitrogen here, etc., etc., etc. So what I obtain is a binary vector of properties of the molecule, and my hope is that this binary vector is going to capture the overall properties, chemical properties of the compound. And this has been the cornerstone, I believe, of chemoinformatics for many years. For instance, fingerprints is what you use for comparing molecules and assessing whether two molecules are similar or not, or fingerprints can be used as input for any machine learning tasks, right? Because those are numerical features of the compound. However, we are in the era of big data, so we actually know much more about the molecules than just the chemical structure. For instance, if I do a search for this molecule, in public databases, I will soon realize that this molecule is called phenanthrene. I know people have suggested it as an antimalarial. They even know the mechanism of action, and they even know some off targets in the host and even some side effects like cardiotoxicity, right? So at some point we asked, wouldn't it be nice to, on top of the chemical information, to actually capture all of this information that we have for small molecules? And this is what we do in this arrow here, which is the chemical checker indeed. It's trying to convert all the information that we can gather for any compound beyond chemistry into a numerical vector that is as useful as the fingerprints used to be. All right, so this is what basically the main asset of my postdoc days, where we developed this huge database of pre-computed, we call them chemical checker signatures, that are able to capture all of the information that we can find in the public domain for each of the compounds that are listed in the public domain. Okay, so this is just a very small chunk of the database. As you can see here in the rows, different molecules. I think I'm just plotting a few hundreds, and you can see here in the columns different numerical values. And each section here corresponds to a part of the, we call it bioactivity space, that we are aiming to capture. So the classical solution has always been to represent numerically the chemistry of the compounds, the chemical properties of the compounds. With the chemical checker, we are aiming to do exactly the same for the target profile of the compounds, for the quote-unquote network profile of the compounds, whatever that means, we will dig into it now, for the cellular profile or the phenotypic profile of the compounds, and even for the clinical profile of the compounds when we have it. Okay, and then I'm just organizing here or stratifying here these compounds by different therapeutic categories, and I think it's quite apparent that depending on the category, we can observe already some patterns, which is indicative that these signatures might have some relevant information within them. Okay, so this is, as you can imagine, a massive effort of data integration, and the way we kind of guide this data integration effort is through biomedical knowledge graphs. I don't know if you are familiar with knowledge graphs in general. The idea is that this is a convenient database structure that we can use to allocate the bioactivity information that we can find in the public domain. This is the knowledge graph as we have it today. Here in the middle, you can see genes. Then you can see that compounds interact with genes. You can see that genes are associated with diseases. You can see that diseases are localized in tissues. You can see that cells are part of tissues. Genes belong to pathways. They have functions. They have domains, etc., etc., etc. So the knowledge graph captures all the relationships that we believe make sense biologically. And then what we do once we have defined this structure is we mine the public domain for evidence of interactions. So here you can see just a very small snapshot of the knowledge graph. The knowledge graph that we have as of today contains about 70 million edges. Here I'm just showing 50,000 of them. And you can see that there are some compounds that interact with genes. We see genes that are actually correlated with diseases, etc., etc., etc. Right? So this is the entry point that we have for building these chemical checker signatures that I was mentioning. Of course, this we can do it for any entity, but our focus today is compounds. So what we are actually aiming to do is to build signatures or embeddings or numerical representations of the nodes that we can find here. Okay. Good. So as I said, this has been a major data integration effort. Thankfully, today everything is kind of automated, and every 6 months we make updates of the chemical checker. The database that we have in the public domain today contains about 1 million bioactive molecules belonging, and we are stratifying these signatures in 25 data types that, as I said before, they go in order of increasing complexity from chemical data up to the clinics, going through targets, networks, and cells. You can find the chemical checker in these web pages. And overall, we have developed this resource, and now we are testing it in many scenarios. For instance, 2 years ago, we tested the chemical checker in a Dream Challenge competition, and we were amongst the winning teams. So it's certainly something that has some predictive capacity, and we are trying to exploit it in different scenarios. Which scenarios, or what can you do precisely with the chemical checker? So the first, or one of the first examples that we wanted to showcase was this idea of doing similarity searches that go beyond chemical similarity. Similarity searches that capture not only the chemistry of the compounds, but also the bioactivity profiles of the compounds. And here I'm showing an example that I like. I hope you're familiar with this type of 2D representations. This is basically a PCA. It's a type of plot called TSNE, where 2 points are close by if they belong. 2 points are 2 molecules, and 2 molecules are close by if they have similar signatures. Yeah, and I'm plotting here 3 maps. The map that corresponds to the transcriptional response of compounds. Yeah, so here 2 compounds that are nearby in this map. This means that they have a similar transcriptional response in cells. In the second map, I'm showing compounds that have similar cell sensitivity profiles. This means that if we test these 2 compounds across a panel of cell lines, the cell lines that will die are similar. And finally, I'm just showing a chemical similarity map where if 2 compounds are nearby, they are similar. If they are not nearby, it means they are different. So in this case, I'm mining, or I'm looking for compounds that have similar, let's say, phenotypic responses. Cells respond similarly transcriptionally. The cells that die in a cell sensitivity profile are pretty much the same. But when we look at the chemical structure of these compounds, they do have very similar structures. Yeah, you can see the structures here. They certainly are not analogs among each other, even if they have very similar phenotypic effects. One of the molecules here was uncharacterized. Indeed, all of these molecules, it's known that they interact in one way or the other with the DNA. And this might explain perhaps why do they have similar phenotypic responses in cells. We can also do the opposite. We can look for compounds that look very similar chemically. For instance, here in orange, I'm plotting compounds that belong to the family of cephalosporins. You might know this family. It's a famous antibiotics family. Yeah, so I have different analogs of cephalosporins, and in purple, I have different analogs, different sulfonamides. Yeah, so obviously, when I map different instances of these families in the map, they are clustered in the chemical structure space. In the mechanism of action, they are also clustered. Indeed, all the members of these families, they interact with the same targets. But then when we look at the transcriptional profile in cells, we observe that the profile is very scattered. This means that the response of cell lines to different members of these families is very variable. And this might explain why some of these antibiotics have for instance, more side effects than others, okay, in the host cells. All right, other things that we can do following like with this chemical similarity concepts that I was mentioning. One thing that we can do also is to try and go beyond only small molecules and start considering antibiotics. And the exercise that I'm suggesting here is the following. We have an antibiotic drug, daclizumab in this case, that is basically an antibody that interacts with the IL-2 receptor. We can go in the databases and collect the transcriptional profile of daclizumab, and I'm plotting it here. Yeah. So now that I have a transcriptional profile of daclizumab, which is an antibody drug, I can look for molecules in the chemical checker that have similar transcriptional profiles. For instance, you can see this first molecule here. It has a very similar pattern of transcription. And then I can make the hypothesis or the educated guess that this compound might have a similar phenotypic effect to daclizumab, right? Might be an analog or a mimetic of daclizumab. This is what we did. We screened about 10,000 compounds for their similarity to the signature of daclizumab, and we came up with 19 significant hits. Meaning 19 molecules that had a significantly similar profile to the profile of daclizumab. And we tested all of them. And of the 19 that we tested, 14 worked to some degree. Okay, here I'm showing some examples. You can see here the IL-2 dependent proliferation. You can see here what happens with daclizumab. And when we expose different compounds, I'm plotting here 4 of them, you can see also a dose-dependent response that is similar to the one exerted by daclizumab. Interestingly, because the comparison here is not done at the target level, but at the transcriptional level, it might be that compounds do not interact precisely with the IL-2 receptor, but somewhere else in the IL-2 receptor signaling pathway. And actually, this is, we believe that this is the case. We found some compounds that interacted with the phosphorylation state of STAT5, which is downstream the pathway, while some others didn't, which suggests perhaps that these compounds are interacting downstream the pathway in 2 different branches. The other thing we can do with these chemical checker signatures is the complete opposite of looking for mimetics. We can actually look for reversion of signatures. And in this case, we can focus on disease signatures. For instance, we collected disease signatures for Alzheimer's. Yeah. What do we mean by disease signatures for Alzheimer's? We had in our laboratory mouse models of the disease, and we also had some cellular models of the disease, and we collected gene expression profile for these models. Yeah, so we have signatures of genes that are over and underexpressed in the disease state compared to the healthy or wild type. And then we can screen the chemical checker for molecules that have not the similar transcriptional profile, but the reverse transcriptional profile, like this case, right? Where genes that are underexpressed, they are overexpressed by the compound, and genes that are overexpressed in the disease, they are underexpressed by the compound. The hypothesis, of course, is that by finding such compounds, we will be able to cancel out the disease signatures and hopefully bring the model back to a healthy-like state. Yeah. So we did that as part of a very large computational screening that involved many other tests. Today, I'm just going to focus on what I've just presented, which is this reversion of transcriptional signatures. And in particular, I will focus on the cellular models of Alzheimer's that we developed, which basically harbor familial mutations of AD. We collected gene expression, and as I said, we just do a virtual screening, a similarity search for reversion. Yeah, so a reversion search. We did that. We identified, in this case, 3 strong compounds that we tested in the laboratory. These compounds are noscapine, palbociclib, and another drug, and they have not been tested for Alzheimer's. And then what we basically asked is whether these compounds were indeed reverting the signature as expected. So here what I'm plotting are genes that are thought to be relevant for Alzheimer's. They state their expression state in the Alzheimer model and what happens to their expression when they are exposed to the compound. So for instance, BIN1 was underexpressed in the Alzheimer model. When we treat it with noscapine, the expression goes more to a wild type like or more to a basal level. For CDC25A was overexpressed in the Alzheimer's model and goes down to a basal-like state when they are exposed to noscapine. This is not anecdotal. This actually happens for many of the genes that are over in blue and under in red expressed in these cells. Okay, there's of course still a long way to go for these drugs to become actually Alzheimer's drugs, but at least the principle that we were able to revert transcriptional signatures was demonstrated here. Okay, so I hope this sounds interesting and useful to some degree, but I'm sure that many of you have the burning question. Well, this is interesting, but the chemical space is huge. We know the chemical space is huge, it's unattainable. And we know that actually the chemical space that we have characterized and the chemical space that is available in the public databases is actually very small and very narrow. So we are facing a problem that I like to make this analogy with the problem that the Romans had when they trying to map the world, right? They had a very good understanding or kind of good understanding of the Mare Nostrum, the Mediterranean Sea, because they had been exploring it all the time over and over. But when it comes to the outer world, the more interesting parts of the world, they actually didn't know anything. Yeah, and this is precisely the problem that as modelers in chemoinformatics we face. And certainly with the chemical checker we face this problem. We are kind of good in getting good signatures, precise signatures in regions of the chemical space that are already well explored. But when we have to extrapolate, things become much more difficult and challenging. So a more honest view of the chemical checker, I believe, would be this one, where we always have chemical information, of course, because chemical information is almost always available for many compounds. But then when it goes to target information, we start losing a lot of data. Certainly we lose a lot of data when we try to gather phenotypic profiling data. And certainly we lose a lot of data when we try to gather information from the clinics, for the simple reason that the vast majority of the compounds have not been tested in humans yet. Okay. So how do we tackle this huge limitation of applicability of the chemical checker? This has been the work of the last couple of years, actually. And to cut a very long story short, we came up with this strategy based on Siamese neural networks. I don't know if you are familiar with this type of networks. These are networks that work relatively well for future learning or low data scenarios, and they basically leverage the notion of similarity between pairs or triplets of compounds. The idea is that when we want to impute some information in one of the spaces, we gather the information that we have from other spaces to at least assess whether two compounds will be similar or not in a particular space where we don't have information for those compounds based on their similarities in other spaces. Okay, that's qualitatively speaking, this is the concept behind a Siamese neural network like the one we trained. As you can imagine, we have 1 million compounds in the chemical checker. If we start doing pairs of compounds, the problem explodes computationally. And this is why it took quite a lot of time to train such a network. And we had to use supercomputing power for over a year to just train one of these networks. And we trained 25 of them because we had 25 chemical checker spaces. But at the end of the day, we have it. So we have a set of 25 Siamese neural networks that are able to do the following. Given a molecule for which we have incomplete data, these Siamese neural networks are able, in principle, to infer or impute the data where it's missing. Okay, we try not to be naive. We are aware that this is stretching a lot the predictive capacity of the chemical checkers. So we did our best to assess first the accuracy of the molecules, and second of the predictions, and second the confidence. Okay, both things are very important when we're doing machine learning, as you know. You can see when we look like globally at the accuracy of these predictions, you can see that for many of the spaces we get, if we are measuring accuracy in binary terms, let's say similar, not similar, we get perhaps good enough predictive power. For instance, in the binding space, we get quite good prediction. But when we go to spaces that are certainly more difficult, like the transcriptional space, the prediction power globally drops. Okay. Within each of these global measures, we can try to assess the confidence of the predictions. And you can, as you can see here, let's focus on binding again. You can see in blue regions of the chemical space that we believe to be confident. Those are the mare nostrum, let's say, and regions of the chemical space in red that we believe to be less confident. It would be the outer wall. Yeah. And as you can see, here, for instance, in the binding, we have plenty of confident regions, but we have regions that is not so confident. When it comes to transcription, we have plenty of non-confident regions and small region that we can predict with good enough, hopefully, confidence. Okay. The question at the end of the day is whether these imputed signatures are useful or not. So we actually try to do a lot of prospective analysis. For instance, one thing that you can do is you can take all of these signatures. Now it's complete data that we have for any molecule, map them on the 2D space, and see whether this 2D representation actually has some sort of biological relevance. I'm taking here drug molecules, and I'm coloring here by different disease categories. And indeed, you can see that molecules belonging to certain categories are mapped into specific regions of the bioactive space, or the bioactivity space according to the chemical checker. Yeah, oncology drugs map here, infectious disease drugs map here, cardiology, etc. Our goal is to go beyond drugs, of course, which are already quite well studied. For instance, if we take metabolites from different species, you can see also that these chemical checker signatures are able to organize the chemical space in a way that is meaningful, at least at the species level. If within human metabolites, for instance, we stratify by tissues and fluids, you can also see some degree of organization. Or even if we look at food ingredients, you can see that different food ingredients, they map on different regions of this chemical checker space. Okay. How is this relevant for machine learning? Well, our hypothesis is that by using these chemical checker signatures, we are actually performing a sort of a transfer learning exercise, right? Where we have collected a lot of data from the public domain. We obtain a dense representation, numerical representation of the molecules that hopefully captures a lot of the information that we could get from the public domain. And then we depart from here to build our predictor of interest. Yeah, this is what it's called, generally speaking, transfer learning. You train on big data, then you fine tune on your data set of interest. This we have applied, for instance, in the case of HCD, which is, you might know it, is a drug discovery center based in the University of Cape Town. They have a lot of data that they have been collecting over the years for about 13,000 compounds. They've never done modeling on them, right? So we basically traveled there. Together with them, we analyzed the data and we built different predictive models for each of the assays that they have in place, right? So with the idea of building a virtual screening cascade that is complementary to their experimental screening cascade. This is what we did for wholesale assays. They are basically focused on Plasmodium falciparum, but also Mycobacterium tuberculosis and escape pathogens. They also have some cytotoxicity assays. They have some ADMET assays and primary, secondary, and tertiary screens, and also some enzymology assays. So we build models systematically with this system for each of the assays. And now they are testing whether these molecules are helping them reducing the attrition rate. Overall, we've done a kind of an assessment of the performance of the chemical checker-based models. And as you can see here, when compared to the chemistry-only models, there are gains, sometimes substantial gains, when we are using this transfer learning approach or this adoption of data from the public domain to build the specific models. The gain of performance is most notable as we go to the physiologically relevant tasks. Here in orange is not so notable when we stay with the more molecular-based tasks, more the biophysics tasks. Okay, so I would argue that the chemical checker becomes useful if you are actually aiming at more phenotypic or whole cell assays, as opposed to the biophysics assays, for which typically the descriptors that we have already tend to be quite useful. With these signatures, we can even focus. For instance, let's focus here in this benchmark that we have here. This is a toxicity panel. You might know it, TOX21, composed of 12 toxicity outcomes. We have built models one by one, and we are comparing the performance of the chemical checker predictor with a comparable predictor built only on chemical information. And as you can see here, for some of the tasks, the performance gain is substantial. Yeah, so this is really what we want. We want to be able to have a tool that, without putting a lot of extra effort, is going to give us models that we believe are state of the art. We can do more things because we have this concept of signatures and spaces and projections. We can actually map the activities on the chemical space, as seen, for instance, by the chemistry space, the target space here in the B, or the phenotypic spaces in the D. Those are the cell-based assays. And we can see which are the spaces that are able to organize better the active compounds here in black. Okay, so here it's very clear that actually the space B, which relates to high throughput screening in other assays, is actually very useful to get a predictive model for these toxicity outcomes. Okay. Finally, and perhaps this is the most important plot, we can also do some sort of robustness analysis. And by robustness here, I mean what happens if we remove more and more data from the training set so that we simulate a low data scenario. If we do so, you can see that the chemical checker predictors, they of course lose predictive power as expected because we have less and less training data, but they do so at a lower rate than the chemistry-only based predictions. Okay, so I would argue here that, at least based on those benchmarks, these chemical checker descriptors, they give you good predictive ability, and hopefully they will also allow you to perform decent models with comparatively less data, which is of course very relevant. It's certainly very relevant in the global south, where the data are very, very scarce. Okay, so let me introduce very briefly another concept. This is the concept of generative models. In this case, in generative models, as you know, and we will have later a presentation that is going to be more dedicated to that. When we are trying to do generative models, the task is completely the opposite. We have an activity of interest and we want to predict small molecules, chemical structures, drawings that are able to fulfill our activity of interest. Yeah. So the task is more complex. I'm not going to go into the details, but long story short, these chemical checker signatures also allow you to do to run such generative models with just a few tweaks. Here I'm just showing some molecules that we have been generating for this consortium called Open Source Malaria. You can see a moiety here that is kind of fixed. And then you can see some derivatives of these molecules, and the more blue the background, the more active we predict these molecules to be. Right? We generated, I think, about 500,000 compounds that we believe might be interesting for their antimalarial activity. We can visualize this in this tree map and also highlight the regions that we believe to be active. Here in red, we can zoom in and identify the compounds that we might want to synthesize. This is what they did. The team of Matthew Todd in the University College London, they have synthesized some of these compounds, and now they are testing in the lab. Okay, we'll see the outcome. All of these compounds have been proposed by a generative model. Okay, and finally, let me just mention very briefly the other projects that we have, because as I said in the beginning, our mission is to actually make an impact in the Global South, and this means that we cannot focus on one project only. We have to try to diversify our impact in different institutions. So if you go to the website, you will have the chance to get an idea of the types of projects that we are doing. Today, I guess I just want to focus on this project that we are doing in Cameroon that is related to the discovery of natural product-like compounds that might have antiviral activity. The idea or the network that we are trying to build for this, and we haven't started yet, so unfortunately I'm not able to show results, is basically scaffold hopping exercise. And you might be familiar with this concept, the scaffold hopping. What you want to do is you have a seed compound or a base compound. You want to make a different compound that doesn't look similarly in the 2D space. Perhaps it will look similar in the 3D space, but certainly doesn't in the 2D space. This usually involves like changing the scaffold that maintains or even improves the activity. Okay, so this is a bit the setting. You want to maintain the activity of the compound, but you want to change its chemical structure. Yeah, we have assembled already from Campbell a data set of these triplets, that is compounds that are similar on the 3D space, are not similar on the 2D space, and they have similar activities. And now we can use this as a training set for a network that would look like this. Yeah, it will contain triplets of a natural product-like compound on one side, a synthetically accessible compound on the other side, and the assay where the activity is measured on the other side. Okay, so we have a triplet, and we can train a network like this with the idea that basically, in practice, at inference time, we can come with a natural product-like compound. In our case, it's going to be most likely a natural product of medicinal plants, of African medicinal plants. And we want to predict a synthetically accessible compound based on this seed compound that will retain the activity. Okay, that's the framework of the scaffold hopping exercise that we are actually trying to do at the moment. And this project is funded by the Gates Foundation, so it's going to be a long project where hopefully we will be able to test our predictions here. Yeah. So basically, simplifying natural product compounds while retaining their activity. Okay, and I think that with this, I just want to wrap up a few take-home messages. I have presented today briefly Cilia. We are a non-profit organization with the mission of democratizing access of AI and ML tools for biomedical research. We are trying to build an international network of collaborators or beneficiaries. We combine remote working with on-site project development and capacity building. Our assets or everything we do is open source, and we try to basically stay up to date with what we publish. We work at the intersection between academia, as you might guess, also startups and why not pharmaceutical companies. And we are always welcoming contributors, collaborators, and supporters. And that's all. Thank you.
Can we ask some questions?
Yes, of course. Go ahead, Jerry.
So, yeah, I thought it was a very interesting talk. And I'm wondering if you ever use your technology to investigate possible side effects that could occur with these different drugs.
Yes, thank you for this question. Yes.
We cannot hear you.
Oh, apologies. Okay. Can you hear me now?
Yes.
Okay, apologies. Thank you for the question. Indeed, we have built, I would say, a list of around 25 models that are aim at predicting the side effects of compounds. And the types of tasks that we have typically are at the disease category. Yeah, so it's not the specific side effect, but some sort of aggregation of the potential side effects. And even we have like more general models that basically try to predict whether a compound is going to be toxic or likely to fail in phase 1 of clinical trials or not. Yeah, but we haven't tested anything, of course. Yeah, so I think that there's still a long way to go before getting competitive predictors here.
Yeah, you would need human tests for that. So, that's not something easy to do. Yeah.
Yeah.
Thank you for your excellent presentation. Dr. Medina Franco asked, for the student, what is your advice to students who's working in a non-profit organization?
My advice for students who are working in a non-profit organization, this is the question, right?
Yes.
Yeah. So in many ways, it very much looks like being in an academic group, right? Because, for instance, we aim at publishing articles, we read articles all the time, and we interact with the scientific community overall. But of course, there's more things we need to do. For instance, we need to think and care about the beneficiaries, and sometimes we need to work in contexts that are not so easy to work in. For instance, we are traveling now to Cameroon, and there the computational capacity is not the same that we get in a country from the global north. Okay, so my advice would be that if you're interested in working in a non-profit organization, first you need to be able to kind of shift between these two worlds, a world where the academic world where resources are kind of okay-ish, and the non-profit world where resources are very scarce, and you need to be comfortable to manage both situations. Of course, you need to care about the mission. The mission is very important. It's not only about publishing scientific papers, it's only about building capacity. And sometimes when you are building capacity, you don't get the same credit than when you are publishing scientific papers. So you also need to be driven by mission, not only by scientific productivity. Yeah. The good part, of course, is that you get to see a lot of different projects, and hopefully you get to see the impact of what you're doing, because you see those groups that you are helping actually grow thanks to the computational assets that you are building.
Okay, thank you, Dr. Miquel. We need to continue with our program. Thank you for your presentation. Our next speaker is Dr. Eli Fernandez de Gortari. Dr. Eli is currently working at the Department of Nanosafety at the International Iberian Nanotechnology Laboratory in Portugal. Dr. Eli is a Tifakin alumni, and we are very happy to hear from him today. Voy a presentar al siguiente invitado. El doctor Eli Fernández de Gortari es egresado de la Facultad de Química de la Universidad Nacional Autónoma de México, donde obtuvo el grado de maestro en ciencias dentro del Departamento de Físico-Química Teórica y el grado de doctor en el área de Quimioinformática aplicado al diseño de fármacos. Cuenta con experiencia postdoctoral dentro del área de diseño de fármacos asistido por computadora en la Universidad de Michigan y en la Universidad de Minnesota, donde desarrolló activadores de la proteína cerca con carácter multiobjetivo como alternativa clínica para el tratamiento de insuficiencia cardíaca. Cuenta con distinciones como la medalla Alfonso Caso, el premio Drug Discovery de la Universidad de Michigan y es miembro del Sistema Nacional de Investigadores Nivel 1. El Dr. Eli forma parte de la Junta de la sección Chemical Information Science de la plataforma F1000. También es autor de diferentes artículos científicos, capítulos de libros y contenido educativo a nivel medio y medio superior. Actualmente se encuentra trabajando en el área de toxicología computacional dentro del grupo de laboratorio de nanotecnología en Portugal, donde aplica técnicas derivadas de quimioinformática, modelado molecular y de aprendizaje de máquinas para la representación, predicción y diseño de nanoentidades. Próximamente, el Dr. Eli se unirá como Senior Science al grupo de quimioinformática liderado por el Dr. Richard Sterhout de la empresa Benevolent en Inglaterra. Bienvenido, Dr. Eli, y muchas gracias por su presencia. La audiencia es suya. Gracias.
Muchas gracias, Fernanda. Muchas gracias. Buenos días. Hello, everyone. I'm gonna give this talk in Spanish, but the talk in English is gonna be available in a link somewhere. So Thank you so much. Voy a empezar a hablar en español. Ok. Bueno, muchas gracias por estar aquí. Mi nombre, como dice Fernanda, es Eli Fernández de Gortari y les quiero hablar un poco sobre el rol que pueden jugar, justamente retomando un poco la plática del doctor Mikel en el sur del planeta, el papel que pueden jugar los modelos generativos para tratar de subsanar los problemas que tenemos sobre acceso a la información para el diseño de fármacos. Bueno, a ver, en general, como quimioinformáticos, lo que queremos desarrollar es una molécula que cumpla con ciertas propiedades y que además tenga afinidad por una o más dianas biológicas que estén asociadas con alguna enfermedad en particular de nuestro interés. Sin embargo, la información que más nos sirve para esto es la información relacionada con ensayos de afinidad enzimática. Aquí les muestro una gráfica que, por ejemplo, en Quimioinformática, una famosa base de datos a la cual tenemos acceso público, solamente cuenta aproximadamente con el 30% del total de sus ensayos sobre afinidad. Realmente es pobre esta información. Y hace poco estuve leyendo un trabajo de Inteligencia, en su grupo desarrollaron una base de datos consenso, donde incluyeron bases de datos como ChemBL, como PubChem, como Binding Database, entre otras, y hicieron un estudio completo cómo poder unir toda la información de afinidad en una sola, y también con todo el proceso de curado que esto implica. Y hablan de que solamente más o menos el 40% de las moléculas se encuentran en una o más fuentes. Aproximadamente solamente 0.14% de estas moléculas están en todas las fuentes. Entonces se imagina que en el momento que uno empieza a buscar información para poder iniciar un proyecto, pues tiene uno que hacer una labor ardua, ¿verdad? Porque puede ser que las bases de datos que estamos revisando no incluyan la información que nosotros estamos buscando. Y solamente el 65% aproximadamente tiene la misma representación como Smile canónica. Y de estas, solamente 306,000 compuestos pueden ser comparados solamente por este tipo de representación. Y solo 387 compuestos están en todas las bases de datos como con la misma Smile canónica. Hablando de diversidad, podemos ver, por ejemplo, los Murco Scaffold, que es una métrica para hacerlo, y podemos ver que solamente el 48% está compartido por todas las bases de datos. Y solamente, por dos o más bases de datos y solamente 0.3 por todas. Ahora, hablando de scaffolds únicos, ¿no? Solamente tenemos 24% en Binding Database, en ChemBL tenemos 26%, en UPAR 22%, y en una que se llama Proof and Drugs tenemos solamente 36%. Solo eso es baja la diversidad y de scaffolds únicos. Y respecto al número total de compuestos obtenidos bajo este proceso de curado, Proofs and Drugs solo tiene una diversidad respecto a este total de compuestos del 52%, y un par del 58%, y el resto está aproximadamente entre el 31 y el 36%. Aquí también les muestro el espacio químico con una técnica de reducción dimensional que se llama UPAT, de 2,000 moléculas que fueron seleccionadas aleatoriamente y representadas con un fingerprint que se llama Extended Connectivity Fingerprint con radio 2. Y podemos ver que, bueno, aunque comparten gran parte del espacio químico, hay algunas que están mucho más, digamos, agrupadas en ciertas regiones del espacio respecto a otras. Esto es para llamar la atención en el sentido que cuando estamos empezando un proyecto es necesario no solamente ver todas las posibles fuentes, sino también tomar ya en cuenta que muchas veces no vamos a encontrar la información que nosotros necesitamos. Otra cosa bastante interesante es el número de targets, bueno, el número de dianas que tenemos a nuestra disposición, el número de datos de compuestos que pueden ejercer un efecto biológico en distintas dianas. Entonces, por ejemplo, tenemos que en PopChem solamente son 3,000, en UPAR 1,500 más o menos, 1,500,000, 1,600. En Proof and Drugs tenemos 4,000, igual más o menos en KMBL, y en Binding Database solo 758 targets. Lo que está pasando aquí es que si tomamos en cuenta que tenemos aproximadamente 60,000 familias de proteínas, pues la cantidad de proteínas a las cuales tenemos acceso sobre datos de bioactividad es realmente bajo. Y otra, hablando sobre Machine Learning supervisado, sobre aprendizaje de máquina supervisado, ¿no? Necesitamos datos muchos datos que estén anotados para poder realizar el proceso de aprendizaje. También necesitamos tomar en cuenta que los datos estén balanceados, es decir, que si por ejemplo vamos a hacer un clasificador que pueda distinguir entre dos tipos de compuestos, por ejemplo activos e inactivos generalmente en esta área, tenemos que tener datos 50% de una clase y 50% de la otra. Hay técnicas para poder lidiar con esto, pero si el desbalance entre estas clases es muy alto, pues el proceso de aprendizaje va a ser muy malo, ¿no? En general no vamos a obtener resultados fiables. Y como pueden ver en este gráfico, en este gráfico que les muestro, la cantidad que tenemos de activos contra activos débiles e inactivos es muy fuerte, ¿no? El desbalance que tenemos. Entonces, esto también es un gran problema para poder usar directamente estos datos en el procesamiento de aprendizaje de máquina. Entonces, hoy les quiero hablar de dos ejemplos de trabajos uno que hice durante mi estancia postdoctoral y otro que recientemente realicé en colaboración con gente de la Universidad Nacional y de la Universidad de Michigan, donde utilizamos generadores para tratar de aliviar un poquito todos estos problemas de los que les estoy hablando. Entonces, el primero es el estudio que hicimos para poder encontrar activadores de una proteína llamada cerca, que es una bomba de calcio que está en el retículo endoplásmico de cardiomiocitos. Esta proteína es importante porque está vinculada con un problema de salud llamado fallo cardíaco, que por ejemplo en México implica 50,000 muertes al año y más o menos en Estados Unidos significa 1 de cada 9 muertes. O sea, es gigantesca la proporción. El costo más o menos, el que tengo es en Estados Unidos, me disculparán, es de $32 billones por año. Y la proxenosis es bastante mala, es de 5 años, más o menos aproximadamente el 50% de los pacientes. No hay terapias dirigidas directamente a este problema, así que solamente se trata de subsanar síntomas periféricos, como puede ser la acumulación de fluidos o la presión alta. Y pues bueno, obviamente hay una necesidad urgente de poder tratar este problema directamente desde sus causas moleculares, ¿no? Bueno, entonces, como les decía, cerca es una proteína que está muy, muy relacionada con este problema porque se dedica a regular el flujo de calcio en cardiomiocitos desde el lumen de la célula hacia el retículo sarcoendoplasmático. No es la única proteína que está relacionada con esto, porque tenemos también los receptores beta adrenérgicos y también tenemos canales de calcio. Para estas dianas sí hay mucha información, hay compuestos aprobados por la FDA que se usan comúnmente, pero tienen muchísimos efectos secundarios. Entonces, se está haciendo un esfuerzo para tratar de encontrar otros targets que puedan aliviar estos problemas. Ahora, el problema de cerca es que tiene muy poquita información. O sea, estamos en una zona del espacio químico donde casi no hay información de actividad. Por ejemplo, uno de los únicos activadores que existe es uno llamado CDN 1163, que fue desarrollado en la Universidad de Minnesota en colaboración con una empresa privada y más o menos tiene una potencia de 26 micromolar, 0.26 micromolar y activa a esta proteína hasta el 40%. También es destacar que esta proteína está mediada por un pequeño péptido llamado fosfolambdano, que pues se dedica justamente a inhibir esta proteína. Bueno, y dado que mi proyecto era encontrar activadores o diseñar activadores para esta proteína y la poca información con la que contaba, pues lo primero que hice fue buscar información en la literatura, ¿no? Directamente. Encontré algunas moléculas derivados de productos naturales y productos naturales, como por ejemplo el ginger oil, la capsaicina y el ácido elágico. Pero cuando hicimos las pruebas en el laboratorio para poder rectificar esta actividad, resultó que no eran activas muchas de ellas, ¿no? Este no fue el caso del ácido elágico, por supuesto, y tampoco del compuesto CDN 1163, lo cual sirvió como punto de apoyo, pero solamente un solo compuesto, ¿no? O dos compuestos. La otra información con la que nos basamos fue información sobre el sitio de unión de fosfolandano, que es una región que se conoce, que regula la actividad de cerca por esta interacción que ya está estudiada. Ciertas mutaciones y estados de protonación en la proteína también afectan la actividad y eso nos podría dar como luz respecto a qué mecanismo podríamos explotar para poder diseñar un activador. Y la otra es que contábamos con una dinámica molecular de 1 microsegundo aproximadamente, que es el tiempo necesario para poder ver cambios significativos en la conformación de cerca que están relacionados con su actividad. Bueno, entonces tomamos esta dinámica molecular y lo que hicimos fue buscar cuáles son las conformaciones más representativas, haciendo un clúster por RMSD. Y estas conformaciones las utilizamos como punto de partida para hacer algo que se llama ensamble docking, ¿no? Que es utilizar cada una de estas conformaciones y en cada una hacer un docking. Utilizamos una base de datos de productos de la medicina tradicional china para hacer esto. Hicimos una comparación de scoring, o del score de la afinidad que nos da el docking contra las distintas conformaciones. Y bueno, elegimos los mejores hits. Después pues, hicimos una muy simple lista de prioridad donde vimos cuáles eran los mejores hits en virtual screening, si estos compuestos estaban reportados de alguna manera en la literatura, fuera funcional o in situ, y después lo probamos experimentalmente y obviamente eso también fue un criterio fuerte. Muchos de los compuestos que están publicados como activadores, que cuando nosotros rectificamos su actividad nos dieron como inactivos, parece que tienen una facilidad muy grande para oxidarse. Entonces, puede ser que estén reportados de esa manera porque no se consideró el estado de oxidación, porque algunos análogos que están oxidados pues sí tienen actividad. A raíz de este proceso encontramos el primer hit, ¿no? Que eso fue sumamente positivo, porque de ahí nos podíamos agarrar para seguir con el proceso. Entonces agarramos estos hits como base para hacer mayor número de análisis. Entre ellos utilizamos una base de datos de la FDA de fragmentos. Entonces son fragmentos que están en compuestos que ya están aprobados por la FDA. Hicimos un virtual screening en el sitio de unión propuesto, utilizando como plantilla, digamos, a los compuestos que encontramos en la fase anterior y pudimos identificar que respecto a las propiedades, distintos fragmentos se unían con mayor afinidad en distintos hotspots del sitio de unión. Esto nos permitió poder diferenciarlos y hacer combinaciones entre ellos para generar nuevas moléculas. Ahora el problema es que había que sintetizarlas, entonces tuvimos que hacer una colaboración con el grupo de química medicinal de la Universidad de Michigan para que nos ayudaran en este aspecto, pero afortunadamente también consideramos la factibilidad sintética para que esta labor no fuera tan ardua ni tan cara, ¿no? Entonces, gracias a esto probamos muchos de nuestros prototipos que formamos con esta metodología combinatoria y obtuvimos más hits. Entonces, esto nos estaba ampliando la diversidad del espacio químico. Ahora, el problema es que si seguíamos por esta ruta, pues nos íbamos a tardar siglos en encontrar algo viable, ¿no? Entonces empezamos a pensar que una estrategia buena era utilizar generadores. Los generadores son una parte de Machine Learning, son un tipo de algoritmo que nos permite aprender una cosa que se llama espacio latente. Este espacio latente es una reducción dimensional de una representación molecular inicial que se logra hacia un espacio continuo que se logra por medio de la recuperación de la representación inicial después de un proceso como de colado, por decir así, y por medio del error de recuperación de la representación inicial se puede generar un proceso de aprendizaje. Es bastante parecido a lo que estamos todos muy acostumbrados con Google Translate, por ejemplo, que puede uno pasar de un idioma a otro. Son algoritmos muy similares, solo que en este caso se usa como representación inicial smiles, por ejemplo, decir español, y se recupera canonical smiles, que podría ser, por ejemplo, inglés. Entonces, al cambiar de representación de entrada y de salida, no solo se logra encontrar la sintaxis de los smiles, sino también, digamos que también puedes encontrar cuáles son las reglas que existen en las moléculas para que sean viables, ¿no? Utilizando esto, pudimos generar un espacio químico considerando la información de los hits que ya teníamos y incluyendo una función de scoring que está acoplada a un optimizador que digamos que busca en el espacio latente los puntos que cumplen mejor con la función predefinida. En esta función incluimos otras dianas, como las que les dije al principio, como los canales de calcio y los receptores adrenérgicos, porque también están relacionados con la función cardíaca. Entonces, lo que queríamos es que no solamente fueran activadores de cerca, sino que también tuvieran actividad contra estas otras dianas. Generamos un número bastante grande de moléculas por medio de este mecanismo que hipotéticamente cumplían con estos criterios, ¿no? De ser multidiana, y también habíamos incluido otras funciones que son importantes en cuestión de propiedades para que esto pueda desarrollar un buen lead molecular. Con estas moléculas entrenamos un clasificador de Machine Learning, Random Forest, por ejemplo, y bueno, en realidad utilizamos muchísimos algoritmos para ver cuál era el mejor, que es uno de los procesos más comunes que se hacen, se selecciona y luego se valida haciendo la comparación entre el training y el test set. Y bueno, generamos un buen modelo y lo utilizamos para hacer virtual screening en una base de datos comercial. ¿Por qué comercial? Porque era mucho más fácil comprarlos en ese momento que mandarlos a sintetizar, además de que es más barato, ¿no? Hicimos este virtual screening en esta base de datos, obtuvimos los hits que nos dijo el modelo y afortunadamente este mecanismo fue útil y encontramos nuevos hits, ahora ya en el orden de nanomolar y que fueron probados funcionalmente y dieron muy buenos resultados, ¿no? Por ejemplo, mejoraron la actividad funcional de cardiomiocitos de pacientes, también disminuyeron el número de arritmias en estas células y o sea que tienen como un efecto protector sobre el corazón, y además resultaron que no son cardiotóxicas. Fueron también probadas experimentalmente. Entonces fue un gran éxito utilizar estos generadores en esta región del espacio que les repito, pues es muy pobre en información. Quiero hoy decirles así como una pequeña introducción de cómo podrían utilizar esto ya directamente. También retomando un poco la charla anterior, es muy importante que simplemente utilicemos las herramientas que ya están publicadas, y muchas veces ya está el código libre para que lo podamos utilizar directamente. Y con muy pocos conocimientos de programación, uno puede empezar a indagar cómo funcionan estos algoritmos y tal vez a la par ir aprendiendo pues un poquito más la teoría que está debajo de ellos, ¿no? Entonces, si ustedes no están familiarizados, por ejemplo, con el entrenamiento de clasificadores, les recomiendo aquí en un link un video que está en YouTube, que es de Data Professor, que les enseña bastantes conceptos de bioinformática, de quimioinformática, pero también les enseña a cómo entrenar y validar un modelo de aprendizaje de máquina clásico para hacer clasificación, utilizando representaciones moleculares, ¿no? También es importante que cuando hagan este proceso siempre chequen métricas para validar su modelo, como por ejemplo Matthews, el coeficiente de correlación de Matthews, el score F o el área debajo de la curva ROC, ¿no? Claro, precisión y demás, la matriz de confusión es importante que lo tomen en cuenta para que estén bien seguros de que su clasificador está bien entrenado. Y además hay una herramienta que se llama Pickle. Hay otras también, pero esta les ayuda a que una vez que entrenaron su modelo, lo puedan guardar para reutilizarlo en otras aplicaciones. Es también sumamente sencillo de utilizar. Y bueno, para empezar así como a platicarles cómo podrían utilizar este algoritmo que les platiqué, este generador, primero tendrían que hacer su clasificador o clasificadores para las dianas que ustedes están interesados y generar una función, aquí les pongo un ejemplo en Python, una función que tome un fingerprint, ¿ok? Y que dé como valor de salida un valor entre 0 y 1, ¿ok? Entonces aquí les muestro, así se define una función en Python, pero pueden revisarlo, o a lo mejor ya lo saben algunos de ustedes. La primera parte de esta función muy simple es tomar el smile que tienen ustedes en su documento y transformarlo en Morgan Fingerprint, ¿no? Que es algo así como el Extended Connectivity Fingerprint que les mostré al principio en el gráfico. Después, pueden, por ejemplo, combinarlo con propiedades físico-químicas. Esto puede ser calculado directamente con librerías que están en un módulo llamado RDKit, ¿no? Es muy fácil también de instalar y de utilizar. Pueden calcular estas propiedades, aquí las muestro en verde, y después hacer una concatenación, o sea, unir la representación del smile con las propiedades y solamente tener cuidado de que a lo mejor tiene que ser escalado, depende del tipo de clasificador, pero todo esto lo pueden aprender con Data Processor. Y una vez que tengan esta representación concatenada, ya la pueden utilizar para pedirle al modelo que ustedes guardaron previamente cuál es el valor o cuál es la predicción, la probabilidad de clasificación que tiene este compuesto en particular. Y bueno, al final, esta pequeña función lo que va a hacer es escupir un valor entre 0 y 1, que ahorita van a ver para qué va a servir. Este paper, que se los recomiendo ampliamente, describe este generador. Este generador es un espacio latente, por decir que ya está preentrenado. O sea, es un espacio continuo donde cada punto representa una molécula y está acoplado a un optimizador llamado Swarm, ¿de acuerdo? Entonces, para instalarlo simplemente se tienen que fijar en las dependencias y tienen que tener instalado una aplicación que se llama Pipe y esto les va a ayudar a hacerlo de forma automática. Aquí les muestro los links para que ustedes tengan acceso al GitHub, al repositorio de GitHub, donde pueden obtener estos dos módulos. Una vez instalado, ahora lo que falta es pues importar algunas de las funciones y algunas de las librerías que nos van a ser útiles a lo largo de esta aplicación. Entonces, por ejemplo, la primera parte son nada más librerías de RDKit, que es un módulo que se usa comúnmente en quimioinformática, en Python, y además algunas librerías que son para poder manejar imágenes, ¿no? Para poder tratar imágenes y que ustedes puedan ver sus resultados gráficamente. Luego, la siguiente parte, en verde, son funciones que están incluidas en este Molecular Swarm que calculan distintas propiedades de las moléculas que ustedes le van a ir dando al generador. Esta parte que está en amarillo es importar el modelo de inferencia, que es el espacio latente, que aquí está marcado como CDDD. Y la última parte es importar obviamente las funciones de los modelos que ustedes preentrenaron. Voy a regresarme tantito, sería la parte inicial, ¿de acuerdo? Entonces lo que van a hacer en esa parte que estaba marcada en rojo en esa diapositiva es importar esta función que ustedes ya generaron con anticipación. Y ya con esto, Lo único que faltaría sería generar una función de score. Esta función de score lo que va a hacer es guiar el proceso de optimización en el espacio latente. Entonces, tiene muchísimas opciones. Ustedes pueden indagar más tanto en el artículo como en la información que viene en el repositorio. Pero bueno, aquí les muestro algunas. Por ejemplo, pueden hacer pueden incluir subestructuras como uno de los criterios de búsqueda en el espacio latente. También pueden poner penalizaciones, por ejemplo, aquí estoy haciendo penalizaciones sobre cadenas alifáticas largas, y también pueden incluir criterios de similitud respecto a una molécula que les interese. Luego va a estar la molécula inicial, que es la molécula como semilla, que en este caso, en el que les platiqué de cerca, pues era uno de los hits que nosotros encontramos. Y finalmente tenemos en verde una cosa que se llama desirability, que en realidad solamente es una manera de poder modular cómo va a responder la función de scoring respecto a las funciones que nosotros le demos como parámetros. Entonces, por ejemplo, si tenemos tenemos una X, ¿no? Entonces, nuestra función, por ejemplo, puede ser el clasificador que nosotros entrenamos previamente. Si el clasificador nos da un valor de 0.01, la función de score en este caso va a responder con un valor de 0. Y así consecutivamente podemos generar una función que se comporte de cierta manera y así poder regular de forma muy precisa cómo va a responder nuestra función de scoring respecto a nuestras funciones parámetro. Y finalmente tenemos en amarillo la función de scoring. Simplemente es una serie de paréntesis donde incluimos todas las funciones como parámetros de esta función de score. Por ejemplo, aquí tenemos otra vez la que estaba definida aquí como QED. Aquí la tenemos, está el nombre, está los valores que va a adquirir respecto a los valores de entrada y luego el peso de este parámetro. Uno puede poner distintos pesos para distintas funciones, ¿de acuerdo? Y pues uno solamente se tiene que fijar que sumen 100, ¿no? Y de esta manera le podemos dar más peso a uno u otro de las variables que estamos considerando en la optimización. Y finalmente, lo único que queda es darle los parámetros al modelo de optimización, donde tenemos que marcar el nombre del compuesto inicial, el número de partículas que vamos a utilizar para el optimizador, el número de optimizadores, el espacio latente, porque además este algoritmo tiene la capacidad de utilizar distintos espacios latentes en caso de que tengan acceso a otro de ellos, si no, el que les mostré ya viene incluido en la paquetería y el nombre de la función de score. Y bueno, finalmente corremos con el número de iteraciones. En esta parte en verde corremos el algoritmo para que realice la optimización y nos va a dar los resultados de esta optimización respecto a la función de score. Y bueno, pueden checar los resultados, ahí están en amarillo, como smiles, o pueden hacerlo visualmente, como se muestra aquí. Esta es la molécula inicial, que les pongo de ejemplo, y esta es la molécula obtenida con la función de score que les mostré anteriormente. Entonces, realmente es fácil de aplicar, ¿no? No es tan complicado. Ahorita lo estoy diciendo muy rápido, pero si a alguien le interesa puede pausar la plática que se quede grabada y puede tratar de hacer esto en compañía con el paper y la información que viene en el repositorio. El otro ejemplo que les quiero dar es la búsqueda de inhibidores de cerca, utilizando generadores. En este caso, empecé con una base de datos de inhibidores, ahí hay un poquito más de información, pero uno de los problemas que hay es tanto la diversidad como que muchos de los inhibidores de cerca tienen muchísimos efectos secundarios. Entonces, se usan generalmente para hacer pruebas experimentales, pero no en ninguna esfera clínica. Entonces, utilicé estos 100 compuestos que están en esta publicación. La mitad de ellos son compuestos inactivos y la mitad de ellos son inhibidores de cerca. Hice el clásico proceso para entrenar un clasificador ¿no? Utilicé la función de score, utilicé distintos mecanismos de clasificación, vi cuál era el mejor, hice las pruebas de performance, etcétera. Utilicé algunas variaciones para poder jugar con el desbalance de la información de esta base de datos y obtuve resultados horrendos, ¿no? Un overfitting que llamamos muy, muy grande. Entonces dije, bueno, aquí hay un problema porque la información con la que cuento es sumamente repetitiva en términos de los scaffolds que están incluidos en esta base de datos. Entonces me puse a indagar y encontré otra paquetería que se llama CREM. Esta paquetería también la pueden encontrar, aquí les muestro, la pueden encontrar en esta publicación que les comparto, y además también está en un repositorio en GitHub, donde pueden bajar el software y utilizarlo directamente. Entonces, bueno, voy a regresar. Lo que hice fue una hipótesis de un SAR continuo. Entonces, este software lo que nos permite es tomar una molécula y basado en una base de datos de fragmentos que está preestablecida, hace mutaciones de las moléculas originales, es decir, cambia fragmentos de la molécula original por otros que son similares, o puede hacer crecimiento de la molécula, ¿no? Adicionando fragmentos, o puede hacer nuevas moléculas haciendo uniones entre fragmentos. Esto me permitió de forma muy conservadora hacer mutaciones pequeñas sobre las moléculas originales para tratar de quedarme en una parte del espacio químico donde todavía se conservara la actividad de las moléculas originales. Utilicé estas moléculas para entrenar nuevamente un modelo y en este caso el rendimiento del modelo y el proceso de aprendizaje pues aumentó muchísimo, ¿no? Y entonces yo pensé, bueno, este ya es un modelo útil. O sea, en este caso la generación de compuestos me ayudó muchísimo a poder entrenar a un modelo que fuera un poquito más útil. Utilicé este modelo para hacer virtual screening en una base de datos de compuestos aprobados por la FDA y encontramos que el algoritmo me decía que las estatinas eran compuestos que seguramente inhiben acerca, lo cual es una pésima noticia, ¿no? Dado que estas moléculas se usan habitualmente en medicina en todos los países muchísimo, ¿no? Y se hicieron pruebas experimentales de estos hits y efectivamente son inhibidores de cerca. Eso ya va a ser presentado en Barcelona pronto por el doctor Cruz Cortés. Y sí, pues sí son inhibidores. Entonces puede ser que muchos de los efectos secundarios de las estatinas, sobre todo a nivel muscular, aquí les muestro en tejido cardíaco y en tejido muscular, puede ser que esté relacionado con esta inhibición. Y después se hicieron dinámicas moleculares para tratar de determinar cuáles son los posibles sitios de unión de las estatinas en cerca. Y bueno, se encontraron varios sitios. Uno es el del fosfolambano, otro es donde habitualmente se unen los inhibidores de cerca. Y también nos dimos cuenta que este modelo era muy bueno para poder determinar, o bastante bueno para poder determinar inhibidores que se unen en la zona BS2, pero no tan buenos para otras regiones, ¿no? Entonces, a lo mejor sería bueno en el futuro generar con otro tipo de información modelos más específicos para los distintos sitios de unión posibles. Y bueno, aquí regresando, por favor revisen esta publicación si les interesa. Aquí están los repositorios donde pueden checar. Y además pueden instalar, si están usando, por ejemplo, Jupyter Notebook, si saben de esto, pueden utilizarlo directamente, pueden instalar directamente CREM, simplemente con este signo de exclamación, pip install CREM, así de fácil es. Y bueno, tienen que importar obviamente las funciones que están en el módulo de CREM que van a utilizar. Por ejemplo, yo aquí estoy nada más importando mutate_mol, que es la función que me va a ayudar a hacer las mutaciones, y algunas librerías que son útiles de RDKit y pandas, ¿no? Para tratar con los archivos de entrada. Luego en la siguiente parte, ustedes ya tienen que tener un archivo con las moléculas de interés y bueno, abrirlo. Les recomiendo siempre checarlo, por eso lo dejé aquí. Hagan como un doble chequeo del contenido de su archivo para ver si tienen todas las moléculas ¿no? Que van a procesar. Y después, miren qué fácil es. Simplemente usas esto que dice aquí, está en la parte de amarillo, dice DB name. Este es el nombre de la base de datos de fragmentos. Pueden usar la que ya está en esta paquetería, en este repositorio, que se llama Replacement 02, o pueden ustedes diseñar su propia base de datos de fragmentos y utilizarla aquí directamente. El radio es qué tan conservador va a ser la mutación. En este caso yo les dije que hice una mutación muy conservadora para tratar también de conservar la actividad de los compuestos iniciales, y simplemente corren esto y pueden elegir el número de moléculas que quieren. Este algoritmo va a generar cientos de moléculas por cada molécula original, pero pueden escoger un número que a ustedes les plazca. En este caso, fueron 20 por cada una de las moléculas que tenían en el archivo inicial. Y bueno, checo de nuevo si está todo bien y obtengo mis moléculas mutadas. Y bueno, espero que esto les sea útil. Cualquier pregunta, está mi correo en la diapositiva inicial. Y pues, tienen alguna pregunta o interés, estoy abierto a contestarlos. Y bueno, eso es todo. Muchísimas gracias.
Muchas gracias, doctor Eli, por la presentación. Yo tengo una pregunta.
Claro, claro.
¿Qué tan comprensibles son las interpretaciones del proceso de toma de decisiones realizado por estos módulos o enfoques de aprendizaje que están en línea? O sea, porque eso quizás es lo que a veces limita a uno el poderlos usar.
Ok, sí. Pues eso depende mucho del autor, ¿no? En general, las buenas prácticas dicen que los programas que uno da al público tienen que estar muy bien documentados, pero realmente lo más común es que no estén bien documentados, ¿no? Son pocos. Por ejemplo, en el caso que puse de CREM, está sumamente bien documentado, tiene ejemplos didácticos, tiene ejemplos de cómo entrenar un modelo de Machine Learning utilizando previamente esta metodología para generar información. Pero en el caso anterior, el que les mostré, el MSO, que es el Swarm, el optimizador Swarm, pues no está tan bien documentado, y por eso también quise mostrar ahorita un poco. ¿Se trabó? No sé.
de la UNAM como investigador asociado C de tiempo completo. El doctor Norberto Sánchez es miembro del Sistema Nacional de Investigadores nivel 1. Ha publicado 16 artículos de investigación en revistas revisadas por pares y un capítulo del libro. Su investigación se centra en la quimiogenómica computacional, particularmente en el desarrollo, validación y aplicación de métodos computacionales para el descubrimiento de fármacos, con especial interés en aquellos basados en inteligencia artificial. Bienvenido, doctor Norberto, y muchas gracias por permitirnos conocer más acerca de su trabajo. Creo que no sé si está silenciado.
Probablemente esté silenciado. Vamos a ver.
Listo, ya tengo audio.
Listo.
Aunque creo que no me ven, pero bueno. Y tampoco tengo habilitada para compartir pantalla.
Creo que en unos minutos te la dan.
En un segundo, el ingeniero Edgar te dará permiso.
Ok, me parece que ya está.
Ya se ve.
Okay, thank you very much for the kind introduction, Fernanda. As he said, I'm Norberto Sánchez Cruz, and today I'm going to talk to you about deep graph learning for protein fragment binding prediction. I will give this presentation in Spanish, but as most of the speakers, all the slides are in English, so non-Spanish speakers can follow the presentation. And I'm going to start now. Please let me know if you can see the slides.
Sí.
Perfecto, cambio para español entonces. Bueno, hoy les voy a hablar sobre el uso de aprendizaje geométrico profundo para la predicción de afinidad entre proteínas y fragmentos. Este es un trabajo que se desarrolló durante mi paso por la empresa de ChemoTargets. Y el contenido de la presentación está organizado de la siguiente forma. Primero les voy a dar una breve introducción, más bien recordatorio diría yo, de qué es el diseño de fármacos o el descubrimiento de fármacos basado en fragmentos. Después les voy a hablar sobre ProSourceScan, que es una plataforma disponible en ChemoTargets con aplicación justamente en el descubrimiento de fármacos basado en fragmentos. Les voy a hablar un poquito sobre cómo surge esta plataforma, los fragmentos que se ocupan, cómo representamos los datos y la aproximación general que se hace. Involucra el uso de dos metodologías principales. Una es la detección de clics y la otra son modelos de aprendizaje geométrico profundo. También les voy a mencionar algunos de los logros que ha tenido esta plataforma y posteriormente entraré en más detalles de cómo están construidos y cómo se constituyen estos modelos de aprendizaje profundo dentro de la plataforma, dentro de lo cual voy a abordar pues, los datos que se utilizan, cómo se tratan, cómo se curan y cómo se generan los modelos. Posteriormente les voy a dar un ejemplo de validación, particularmente en GPCRs, donde voy a hablar pues, sobre cómo construimos los conjuntos de entrenamiento, prueba y validación y cómo se desempeñan ambas metodologías en esta tarea, tanto la detección de clics como los modelos de aprendizaje profundo, para finalizar con algunas conclusiones. Entonces, entrando en el primer tema, que es el descubrimiento de fármacos basado en fragmentos, si lo quisiéramos definir en una sola línea, básicamente podríamos decir que el descubrimiento de fármacos basado en fragmentos es el desarrollo de compuestos potentes o fármacos, con la intención de que sean fármacos, a partir de un fragmento pequeño o de una molécula más pequeña. Esta aproximación tiene sus ventajas y sus desventajas. Las ventajas están sobre todo asociadas al tamaño y a la complejidad de los fragmentos, porque son menores en comparación con moléculas completas, lo cual hace que el espacio a explorar sea menor, porque tan solo las combinaciones de átomos que se pueden dar en una molécula pequeña son mucho menores que las que se dan en una molécula con más átomos. Por la misma razón, es menos probable que contengan grupos que impidan estéricamente la unión a una proteína, y al ser pequeños, pues presentan una oportunidad bastante alta para su optimización. Y dentro de los principales contras que tiene esta aproximación es que a menudo los hits que se encuentran, es decir, los fragmentos que se identifican inicialmente, tienen una afinidad muy baja por las proteínas de interés y además son poco selectivos, pues se unen a más de una proteína. Es decir, la unión no es específica. Bueno, con estos pros y estos contras podemos hablar de algunos casos de éxito, por ejemplo, el lemurafenib, que es un inhibidor de BRAF, una sinasa, el cual fue desarrollado a partir del descubrimiento de que el 7-azindol, señalado ahí en el círculo rojo, se une a BRAF y posteriormente fue optimizado a la molécula que está disponible comercialmente, que es la que se muestra en la imagen. Otro ejemplo exitoso, del cual hay incluso evidencia estructural dentro del Protein Data Bank, es el descubrimiento de inhibidores irreversibles de la fosfolipasa A2, de la fosfolipasa asociada a lipoproteínas A2. En este caso, se descubre que un fragmento de 183 daltons, es decir, un fragmento muy pequeño, se une irreversiblemente a esta proteína con una potencia de 2.77, ¿no? Medida como IC50. Entonces, posteriormente, esta molécula se optimiza y se consigue una molécula con un peso molecular de 614 daltones, es decir, ya una molécula más parecida a lo que actualmente se conoce como un drug-like, una molécula parecida a un fármaco, y cuya potencia es 5 órdenes de magnitud mayor a la del fragmento inicial. En este caso hay estructuras cristalizadas de tanto del fragmento como de la molécula completa, y aunque no se alcanza a ver muy bien, el fragmento está unido en esta sección, mostrado en blanco, y la molécula completa pues se diseñó para que abarcara más parte de la cavidad. Entonces, en resumen, el diseño de fármacos basado en fragmentos funciona. Pues ahora les voy a hablar un poco sobre la plataforma ProSurfScan, que hace referencia a Protein Surface Scanner, que es la plataforma propia de ChemoTargets, que se utiliza para escanear la superficie de una proteína y buscar los fragmentos que se pueden unir a las distintas regiones de esta. ¿Cómo nace esta idea? Bueno, como ya habrán notado, esto se trata de una aproximación basada en estructura. Y lo que queremos hacer es aprender de las estructuras que ya se tienen, estructuras de proteínas unidas a ligandos. Y el recurso por excelencia de este tipo de información es el Protein Data Bank, el PDB. En términos numéricos, el PDB incluye más de 100.000 estructuras de proteínas que están unidas a al menos un ligando, para sumar un total de más de 250.000 posibles sitios de unión. Si analizamos esto en términos de los ligandos, podemos identificar que hay más de 50.000 ligandos diferentes, pero de estos, más de la mitad, cerca de 28.000, ocurren únicamente una vez en toda la base de datos. Por lo cual, si queremos hacer un análisis estadístico o aprendizaje estadístico de las características que tiene que tener la superficie de la proteína para unirse al ligando, pues lo podríamos hacer con muy pocos. Si pusiéramos un punto de corte, por ejemplo, en aquellos que ocurren en más de 50 ocasiones en la base de datos, pues nos quedaríamos únicamente con 484 ligandos, lo cual sería muy poco representativo de todas las moléculas que podríamos abarcar o que podríamos estudiar. Pero si en lugar de analizar los ligandos en su totalidad, analizamos los fragmentos que lo constituyen, en este caso los números cambian. Y entonces si analizamos ahora en términos de fragmentos cuántos hay dentro del Protein Data Bank, encontramos que hay más de 640.000 fragmentos diferentes. Aunque el patrón se repite y más de 390.000 ocurren una sola vez, en este caso, si analizamos cuántos ocurren más de 50 veces, ya tenemos más de 4.700 fragmentos, a lo cual ya es un número 10 veces mayor al número de ligandos que podríamos estudiar, y con la ventaja de que estos fragmentos ocurren en más de un ligando. Ahora, ¿cómo generamos estos fragmentos? Pues para generar estos fragmentos de PDB, lo que utilizamos es un algoritmo que se llama BRICS, que es Breaking of Retrosynthetically Interesting Chemical Substructures. Este algoritmo lo que hace es utilizar 16 reglas de corte, en la cual se identifican 16 tipos de enlace específicos en las moléculas y estas son fragmentadas en ellos. Como un ejemplo esquemático, tendríamos esta molécula que consta de 3 fragmentos diferentes, representados ya sea por color o por figura distinta y unidos por 2 enlaces que podríamos romper. En un primer paso, lo que haría el algoritmo de Briggs sería romper alguno de los 2 enlaces que se pueden romper, y si nos quedamos con el fragmento más grande que se genera de esa ruptura, tendríamos estos 2 que se presentan aquí en la pantalla. En un paso siguiente, se romperían todos los enlaces posibles, en este caso son solo 2, y obtendríamos cada uno de los constituyentes, de tal forma que al final este ligando nos generaría, o esta molécula nos generaría 6 fragmentos diferentes, con el más grande siendo la molécula completa y con los más pequeños siendo cada uno de sus constituyentes. Y en el medio pues tenemos las combinaciones de estos. Ahora, ¿cómo pretendemos con ProcessScan aprender de las características que definen la unión de estos fragmentos a la superficie de una proteína? Pues para esto lo que hacemos es primero identificar el sitio en donde está unido el ligando en cuestión. En este sitio se construye la superficie triangulada de la proteína. Esta superficie triangulada está constituida por un montón de pequeños nodos, que son los vértices de cada triángulo, que están asignados o que son generados a partir de un átomo de la estructura de la proteína. Por lo cual, se les pueden asignar algunas características químicas de estos átomos. Entonces, si trabajamos con todos los puntos, con todos los vértices de esta superficie triangular, tendríamos una representación muy fina de la superficie de la proteína. Entonces, lo que hacemos en lugar de quedarnos con todos los nodos o con todos los vértices es agruparlos de acuerdo a aquellos que estén cerca, es decir, aquellos que compartan una característica, ya sea hidrofóbica, aromática, que vengan de un átomo que puede aceptar o donar puntos de hidrógeno, etcétera, se van a agrupar en una serie de centroides, es decir, estos puntos que están señalados con colores aquí representan a un grupo de vértices de la superficie triangular. Con esto, pues ya nos olvidamos por completo de la superficie y estaríamos representando al ligando o a la molécula en cuestión por estos puntos con características señaladas aquí del lado izquierdo que fueron identificadas previamente. Una vez que hacemos esto, la molécula en cuestión se fragmenta y los puntos con características que estén más cerca de cada uno de los fragmentos son asociados a ellos. Entonces, de esta forma, una vez que tenemos ya los puntos de características asociados a un fragmento, a lo cual me estaré haciendo referencia como el entorno del fragmento, con esto podemos construir un grafo completo. Un grafo completo es un grafo en donde todos los nodos están conectados con todos los nodos. Y en este caso, cada nodo o cada vértice pues va a ser uno de estos puntos de características que va a estar conectado a cada uno de los otros por un eje o arista que va a tener también un descriptor, en este caso conocido como peso, y el cual particularmente para esta aplicación pues va a ser la distancia que lo separa de otro nodo. De tal forma que nuestra representación al final sería algo así. Una serie de nodos unidos todos con todos y cada uno con una característica química diferente. Ahora, con esta representación de la superficie, pues lo que hacemos es olvidarnos ya un poco de los fragmentos. Y los algoritmos que utiliza ProcessScan son básicamente 2. Uno es la detección de clics y el otro son modelos de aprendizaje geométrico profundo. Entonces, ¿cuál es la idea general? Pues la idea general es que si tenemos un grafo de interés, en este caso sería un entorno de alguna proteína que a nosotros nos interese y que no sepamos qué fragmentos o qué ligandos se unen a ella, lo que podemos hacer es compararla con todos los entornos conocidos de la base de datos del PDB. Esta comparación se hace haciendo un producto de grafos y sobre el resultado de este producto, se aplica un algoritmo de detección de clics que es equivalente a buscar el máximo subgrafo común entre el entorno que me interesa y los entornos que conozco. Entonces, al hacer la detección de este máximo subgrafo común, si la similitud es lo suficientemente grande o si el subgrafo es lo suficientemente grande, podríamos decir que el fragmento asociado a este entorno es compatible con el entorno que me interesa. Esto es por el lado de la detección de clics. Por el lado de los modelos de aprendizaje geométrico profundo, lo que se hace es pasar este entorno de interés por una serie de modelos de aprendizaje profundo que son entrenados a partir de los grafos conocidos o de los entornos conocidos de los fragmentos presentes en el PDB. Y la finalidad al final es la misma. Lo que estos modelos me van a predecir es si el fragmento en cuestión es compatible o no con el entorno dado. Esta es la aproximación general. Ahora, cada uno de los algoritmos presenta sus características propias que los hacen aplicables a distintos escenarios. Por ejemplo, la detección de clics nos permite hacer una comparación exhaustiva contra todos los entornos de fragmentos que están presentes en el PDB. Una de las grandes ventajas es que podemos comparar grafos con diferentes órdenes, es decir, con diferente número de nodos, lo cual nos permitiría escanear o comparar ya sea un sitio de unión en particular o incluso toda la superficie de la proteína. El problema es que a medida que es más grande la superficie que queremos escanear, pues la complejidad computacional también crece mucho. Y otra de las grandes ventajas es que si hacemos la detección de los nodos que son compartidos entre un entorno dado y un entorno conocido, podemos asignar el fragmento del entorno conocido al entorno dado haciendo la rotación correspondiente, por lo cual podemos estimar el modo de unión del fragmento a partir del clique que se identifica. Por el otro lado, los modelos de aprendizaje profundo pues solamente están disponibles para fragmentos de los cuales tengamos suficiente información estructural, o sea, suficientes entornos de los cuales aprender. En este caso, nuestras predicciones van a ser realizadas únicamente en grafos con órdenes predefinidos, es decir, se espera que un fragmento se una siempre a entornos de más o menos el mismo tamaño, lo cual hace que la complejidad computacional de su cálculo sea bastante más baja en comparación con la detección de clics, y que además nos permita estimar directamente la compatibilidad de los modos de unión que vienen de simulaciones moleculares, como el acoplamiento molecular o la dinámica molecular. A final de cuentas, estos dos algoritmos embebidos dentro de la plataforma ProcessScan nos han permitido estimar la compatibilidad y el modo de unión de fragmentos a proteínas, lo cual ha resultado en la identificación de compuestos novedosos en diferentes diseños de fármacos. El logro más grande probablemente sea que es que gracias a esta plataforma se pudo identificar la primera molécula que llegó a fases clínicas para el tratamiento de la enfermedad de Huntington. Además de eso, dentro de la empresa hay algunos proyectos propios relacionados con cáncer y con enfermedades respiratorias y proyectos para los cuales la empresa ha sido contratada por empresas externas relacionados con la neurodegeneración, que es el caso de la enfermedad de Huntington, que ya llegó hasta el candidato clínico, y otros relacionados con la inflamación, en donde pues, se tienen avances donde se han identificado ya hits y algunos ya han pasado a la etapa de optimización. Entonces, como desafortunadamente no puedo hablar de estos datos porque son confidenciales de la compañía, lo que voy a hacer es presentarles un ejemplo de aplicación en datos que ya se conocen y que están disponibles públicamente. Entonces, para empezar, ya vamos a entrar de lleno con el aprendizaje geométrico profundo. Entonces, lo primero para generar estos modelos, como ya mencioné, es tener suficientes datos. Entonces, ¿para qué fragmentos tenemos suficientes datos? Bueno, si hacemos un primer filtrado de todos los entornos y todos los fragmentos que tenemos disponibles del PDB, lo primero que hay que hacer es quitar pares proteína-fragmento que sean redundantes. ¿A qué me refiero? A que si en una misma estructura cristalográfica tenemos 8 cadenas que son iguales y que todas están unidas de la misma forma al mismo fragmento, pues esto en realidad es información redundante, que para entrenar un modelo pues no me sirve demasiado. Entonces esto es algo que hay que evitar. Como ya mencionaba, necesitamos que los datos sean suficientes, por lo cual nos quedamos únicamente con fragmentos que ocurrieron al menos 50 veces dentro del PDB, ya quitadas las redundancias. Además de esto, añadimos un par de criterios más, que es que el fragmento estuviera presente en al menos 2 familias de proteínas distintas y en al menos 2 ligandos distintos. Con esto, identificamos más de 4.700 fragmentos únicos, con el problema de que por el algoritmo de fragmentación que utilizamos, ocurren 2 cosas. Los fragmentos más pequeños que se pueden identificar son átomos, como carbono, nitrógeno, oxígeno, y los fragmentos más grandes, como ya había mencionado, son la molécula completa. Entonces, esto en realidad es información que no nos interesa mucho para proyectos basados en fragmentos, así que lo que hicimos fue añadir criterios que se usan en bibliotecas para hacer screening comercial de fragmentos, que son básicamente estos, ¿no? Criterios de fragment likeness. Entonces, nos quedamos con fragmentos que tuvieran un peso molecular menor a 300, menos de 3 aceptores y 3 donadores de puentes de hidrógeno, un log P entre menos 2 y 3, que tuvieran entre 1 y 4 anillos y que tuvieran menos de 3 enlaces rotables. Con esto, el número bajó a 420 fragmentos únicos, que son para los cuales podríamos generar modelos. Pero vemos aquí los fragmentos más pequeños de los 420 que nos quedamos y los más grandes. Entonces, definitivamente ya nos quitamos el problema. Los fragmentos más pequeños ya no son átomos y los fragmentos más grandes definitivamente ya no son moléculas completas. Y lo que se muestra abajo de cada uno de ellos es cuántas veces ocurren dentro de la base de datos. No, mentira, es en cuántos ligandos diferentes ocurren dentro de la base de datos. Ahora, viendo estos números, podrían pensar que les estoy tomando el pelo y paso diciéndoles que poder analizar cerca de 400 compuestos es muy poquito y ahora estoy analizando 400 fragmentos. Sin embargo, aquí lo importante es que estos fragmentos están presentes en muchas moléculas. Entonces, ¿qué tan representativos son estos fragmentos en las bases de datos que conocemos, ¿no? Que tenemos disponibles en el dominio público? Es decir, ¿cuántos compuestos contienen al menos uno de estos fragmentos? Pues si analizamos más de 9 millones de compuestos disponibles en Sync20 que están listos para su compra, vemos que más del 80% contienen al menos uno de estos 420 fragmentos. Y si hacemos el mismo análisis en la última versión de QMBL, que son más de 2 millones de compuestos con actividad biológica reportada, vemos que más del 70% de ellos también contienen al menos uno de estos 420 fragmentos. Es decir, estos son bastante representativos de la información química que se conoce y de la cual se dispone públicamente. Ahora, entrando ya de lleno al aprendizaje geométrico profundo, ¿cómo lo podríamos definir? Pues igual, en una sola línea, podríamos decir que el aprendizaje geométrico profundo consiste en el uso de redes neuronales profundas que son capaces de aprender de datos estructurados en forma de grafo. Como ya habrán notado con lo que mencioné en las diapositivas previas, pues es el caso que tenemos. Tenemos entornos de proteínas representados como un grafo, así que el problema ya está planteado para que podamos usar Deep Graph Learning. En particular, los modelos que nos interesaron aquí son modelos de redes neuronales convolucionales que actúan sobre grafos. Entonces, ¿cuál es la idea general? Pues la idea es que si tenemos un entorno de una proteína, un sitio de unión, o su grafo completo que lo describe, tenemos un vector que describe las características de cada nodo. En este caso, se planteó como un vector binario de 6 posiciones, donde cada una de estas puede ser un 1 o un 0, dependiendo de si presenta o no la característica en cuestión, las que ya había mencionado, hidrofóbico, aromático, aceptor, donador, etc. Y cada uno de estos ejes tiene un peso. En este caso, en lugar de utilizar la distancia, utilizamos una versión escalada del inverso de la distancia. Y en un momento entenderán un poco por qué. Entonces, sobre esto, sobre esta descripción inicial, aplicamos la operación de convolución. ¿En qué consiste la operación de convolución? Pues consiste en calcular nuevas características o nuevos descriptores para cada nodo, que es algo que se conoce como el estado oculto, el hidden state, que se calculan a partir del mensaje que reciben de los nodos adyacentes, es decir, de los nodos que están conectados. En nuestro caso particular, la implementación que usamos es la que se describe en esta ecuación, donde tenemos que para cada estado oculto del nodo se va a calcular a partir de las características propias del nodo más una contribución de las características de cada uno de los nodos que tiene unidos multiplicadas por el peso de los ejes. Entonces, esta es la razón de que los pesos se hayan calculado de esta forma. ¿Por qué? Pues porque para darles un mayor peso a los nodos que estuvieran más cercanos entre sí, que son los que, si lo vemos desde un punto de perspectiva física, tendrían una mayor interacción o una mayor aportación a la interacción con el nodo vecino. Entonces, al aplicar esta transformación, lo que tenemos es esencialmente el mismo grafo, pero donde cada nodo ya recibió el mensaje de todos los nodos adyacentes y ahora sus características son completamente diferentes a las iniciales, porque ya tienen información acerca de su conectividad con los otros nodos y de las características de los otros nodos. ¿Vale? Entonces, al final, lo que hacemos es construir un modelo de clasificación binaria para cada uno de los fragmentos posibles, donde la idea es que para entrenar los modelos partimos de los datos en forma de grafo de los entornos asociados a fragmentos. Esto se hace pasar por un par de capas de convolución, definida como ya lo presenté en la diapositiva anterior. Puesto que cada entorno contiene un diferente número de nodos, tenemos que añadir lo que está aquí marcado como una pooling layer, que lo que hace es básicamente combinar la información de todos los nodos para representar de forma esquemática lo que sería un supernodo que ya contiene la información de todos los nodos del grafo. Y este nodo ya tendría la misma dimensionalidad para todos los grafos. Después de esto, lo que se hace es pasarlo por una red neuronal, una capa lineal de una red neuronal, que es una capa sencilla, una capa normal de las que usan todas las redes neuronales, no las más básicas, para hacer la clasificación, de tal forma que cada uno de estos modelos lo que va a hacer es responderme a la pregunta, es de si el grafo de entrada, o en este caso el entorno de la proteína, es compatible o no con un fragmento, y me devuelve las probabilidades de que lo sea y de que no lo sea. Y ahora, el ejemplo de validación en GPCRs. Ok, pues lo que hicimos aquí para mostrar un ejemplo de aplicación de estos modelos es seleccionar un conjunto de validación primero que consistía en todos los fragmentos que están asociados a GPCRs dentro del Protein Data Bank. Entonces, en este caso, identificamos 290 fragmentos que corresponden a 62 fragmentos únicos. Es decir, hay fragmentos que se unen a varios GPCRs, lo cual no es para nada extraño, ¿no? Para entrenar y probar los modelos, tomamos únicamente fragmentos de proteínas no relacionadas. ¿Y a qué me refiero con proteínas no relacionadas? Pues son proteínas que esencialmente no son GPCRs ni comparten nada con ellas. Es decir, que no tienen el mismo plegamiento, no se pueden alinear con ellas, etc. Entonces, tomando únicamente proteínas no relacionadas, nuestros datos positivos para entrenar el modelo en el contexto de cada fragmento, pues son todos esos entornos que vienen de fragmentos idénticos al que queremos predecir en GPCRs, es decir, a cada uno de estos 62 fragmentos. Y los datos negativos son fragmentos que tomamos al azar de proteínas que no estuvieran relacionadas con los datos positivos para evitar la mayor cantidad de ruido posible y que además mantuvieran una distribución similar en cuanto al número de nodos y a la distancia que mantenían estos nodos. Esto pues para que al final fueran parecidos y representara un reto para los modelos aprender de ellos, poder diferenciarlos. Algo que ya mencionaba Eli en la plática anterior es que un problema que tenemos siempre es el desbalance de datos. Al final, los casos positivos que tenemos siempre son muy pocos en comparación con los datos negativos. Y los datos negativos ni siquiera estamos seguros que son datos negativos, simplemente no sabemos si eso se une o no a las proteínas que nos interesan, por ejemplo. Entonces, si tomáramos todos los datos de proteínas no relacionadas a mi conjunto positivo, el desbalance sería prácticamente una relación de 1 a 100 o 1 a 1000. Entonces, para mantener las cosas no tan desequilibradas, lo que hicimos fue reducir el conjunto negativo de tal forma que se mantuviera un desbalance de 1 a 10 a lo más. De tal forma que por cada caso positivo de un fragmento que se une en el entorno, tengo 10 casos negativos. Y para el entrenamiento y prueba de los modelos hicimos una partición 80-20. Utilizamos 80% de nuestros datos en el conjunto desbalanceado para entrenar al modelo y 20% para probarlo y optimizar los hiperparámetros, asegurándonos que se mantuviera la relación de 1 a 10 en el desbalance, tanto en el entrenamiento como en la prueba. Entonces, ahora, algo que tenemos que usar como referencia es el algoritmo de detección de clics, que también se puede utilizar para este tipo de problemas. Entonces, en este caso, ¿cuál es la idea general? Pues si tenemos un entorno dado, que es decir, estos son los entornos de los fragmentos que sabemos que se unen a los GPCRs, mi conjunto de validación, lo que puedo hacer es búsquedas en los entornos que sabemos que no corresponden a GPCRs, los entornos de fragmentos de proteínas no relacionadas. Entonces, con esto, pues hacemos la detección de clics y ordenamos estos resultados por similitud, es decir, qué tan grande es el clic que se detecta. Y con esto podemos medir cuántos o qué porcentaje de los fragmentos que sabemos que son a GPCRs son recuperados a partir de estructuras no relacionadas. Los resultados aquí se muestran en la gráfica de la derecha. Si tomamos únicamente el mejor resultado de la búsqueda, recuperaríamos el 67% de los fragmentos. Pero si recuperamos los 10 fragmentos los 10 entornos más parecidos, ya recuperaríamos 275 de los 290 fragmentos iniciales, lo cual significaría una recuperación del 95%. Poniendo esto en contexto de las proporciones de cada uno de los fragmentos únicos, tendríamos que para 55 de los 62 fragmentos únicos tenemos una recuperación de más del 60%. Y para mostrarles un poquito cómo funciona este algoritmo, cómo son los resultados que obtenemos, pues dicen que para muestra basta un botón y aquí se los pongo, ¿no? Entonces un ejemplo muy bonito es este, donde se sabe que este fragmento, que es un fragmento de una tirosina, se une a este GPCR. Y entonces este es el que estábamos buscando. Entonces al hacer la búsqueda utilizando únicamente el entorno, encontramos al fragmento dentro de una proteasa de una bacteria que no está para nada relacionada con los GPCRs. Entonces, en este caso, el número de puntos con características que se encontró en el clic fue de 8, es decir, hay 8 puntos de la superficie que coinciden y si se hace el alineamiento entre ellos, lo que obtenemos es esto, es un alineamiento casi perfecto del fragmento, que viene de una proteína que no se relaciona para nada con los GPCRs. Si además vemos la secuencia o más bien los aminoácidos que dan origen a estos puntos de superficie, vemos que, salvo por una fenilalanina, todos los demás son diferentes. Lo cual me dice que tampoco hay una relación por secuencia, ni por plegamiento, ni nada. Entonces, esto es de lo que es capaz la detección de clics. Ahora, si nos vamos por el otro lado y vemos qué tan buenos son los modelos de aprendizaje geométrico profundo, estos modelos fueron entrenados en el conjunto de entrenamiento hasta que no se alcanzara una mejora en el desempeño del modelo para predecir el conjunto de prueba. Esto es algo que se conoce como early stopping, que lo que hace es básicamente evitarme el overfitting de los modelos. Entonces, para esto, los modelos son entrenados utilizando un criterio de optimización, que en este caso es la entropía cruzada, con una serie de pesos. Lo que hace la entropía cruzada está definida aquí en esta ecuación, y lo que hace básicamente es medir el error entre las predicciones y los datos conocidos, asignando un peso a cada una de las clases. En este caso, para balancear, o más bien para contrarrestar el hecho de que los datos están desbalanceados, pues le dimos un peso 10 veces mayor a las predicciones de los casos positivos que a las de los casos negativos. Y entonces lo que vemos en cada uno de nuestros clasificadores binarios son curvas como esta, donde vemos que a medida que avanzamos en las iteraciones en el entrenamiento de la red neuronal, el error va bajando en el conjunto de entrenamiento y seguirá bajando prácticamente indefinidamente. Sin embargo, si analizamos este error en el conjunto de prueba, vemos que llega a un punto en donde se estanca y deja de bajar. Y de hecho, al contrario, si dejáramos avanzar más esto, el error empezaría a subir, que sería el indicativo de que el modelo ya está sobreentrenado y solamente está memorizando los datos en el conjunto de entrenamiento. Y este es el punto ideal en el que hay que dejar de entrenar el modelo y guardar los hiperparámetros. Entonces aquí, donde guardamos los modelos, y son los modelos que se utilizan para hacer las predicciones en el conjunto de validación. Ahora, si analizamos los resultados de los modelos en cuanto a su exactitud balanceada por clases, tanto en el conjunto de entrenamiento como en el conjunto de prueba, lo que vemos es un gráfico como este. Entonces vemos que para 57 de los 62 modelos tenemos una exactitud de más del 60% en ambos casos, tanto en entrenamiento como en prueba, lo cual, poniéndolo en contraste con la detección de clics, nos estaría diciendo que los dos métodos son buenos prediciendo 51 de los 62 fragmentos únicos, con la detección de clics siendo mejor para 4 y los modelos de Graph Learning siendo mejores para 6. Por otro lado, si analizamos la recuperación, es decir, cuántos de los fragmentos que se conoce que se unen a GPCR son predichos bien por los modelos de Deep Graph Learning, lo que vemos es una gráfica así, en donde 38 de los 62 tienen una recuperación de más del 60% en el conjunto de la validación, que a pesar de que pareciera ser poco, es suficiente para recuperar 247 de los 290 fragmentos, lo cual sería una recuperación del 85%. Y además, podemos notar, en general, los fragmentos que presentan una baja recuperación son aquellos que tienen pocas muestras positivas, lo cual nos dice, pues es algo que ya sabemos, ¿no? Mientras más datos tengamos para entrenar a los modelos, mejor van a ser estos datos. En estos términos, pues tendríamos que, en términos de recuperación, o sea, puramente de recuperación, tendríamos que 36 de los fragmentos se pueden predecir bien por ambos modelos, 19 por la detección de clics, 4 por los modelos de aprendizaje geométrico profundo. Entonces, ya como conclusiones finales, lo que presenté en estas diapositivas fue una validación exhaustiva de dos métodos implementados en CrossOverScan para la predicción de la unión entre proteínas y fragmentos. ¿Por qué validación exhaustiva? Porque para esto se utilizaron para validar únicamente fragmentos que saben que se unen a GPCRs, y todos los modelos y todas las búsquedas fueron hechas en proteínas que no están relacionadas a GPCRs, lo cual hay que remarcarlo, porque pues teniendo poca información estructural, obtenemos muy buenas recuperaciones de los fragmentos, 85 y 95%. Entonces, a pesar de que el desempeño general es mejor para la detección de clics, los dos métodos funcionan bien para distintos fragmentos y para distintas aplicaciones, que es algo de lo que mencionaba al inicio, lo cual los hace ser un muy buen complemento el uno del otro. Algo que también hay que remarcar es que el desempeño de estos dos métodos pues definitivamente es mejor si incluimos datos de proteínas relacionadas, donde es mucho más fácil que tengamos superficies o entornos similares para los fragmentos. Y al igual, pues algo de lo que mencionaba al final es que al ser algoritmos que están basados en datos conocidos, en datos estructurales, pues su desempeño va a ser mejor a medida que tengamos más datos experimentales disponibles. No me queda más que agradecer a todo el equipo de ChemoTargets, particularmente a Jordi Mestres, que es el fundador y director científico de la empresa, y a los compañeros que me acompañan en el grupo de diseño de fármacos basado en estructura, que son Adrián, Julia y Xavi Falencas, que es quien dirige el grupo. Y por supuesto a ustedes por su atención. Si tienen preguntas, dudas o comentarios, estaré encantado de escuchar.
Muchas gracias por la presentación, doctor Norberto. Fue demasiado interesante e ilustrativa. Tenemos algunas preguntas. Edgar López comenta: muchas gracias por su presentación, fue bastante ilustrativa. ¿Han pensado en utilizar ProfScan para estudiar receptores o targets huérfanos?
Hola, pues muchas gracias por la pregunta, Edgar, y por el comentario. En realidad sí, pues es básicamente la principal idea de ProfScan. La idea es que sea una herramienta útil para desorganizar estas dianas. O sea, para buscar fragmentos o ligandos en proteínas de las cuales no se conoce nada. Entonces es su principal utilidad, su principal aplicación.
Gracias. Y otra pregunta de Ángel Santiago. Comenta: muy buena presentación, doctor Sánchez. Mi pregunta es para la generación de estos entornos con ProfScan. ¿Es necesario contar con el PDB de un complejo?
Sí, para la base de datos que utilizamos como referencia se basa únicamente en datos estructurales experimentales. Así que todo lo que esté en el PDB está incluido en nuestra base de datos. Para hacer una búsqueda sobre alguna otra diana o alguna otra proteína, se puede hacer ya sea con estructura cristalográfica o con modelos computacionales. De hecho, ahora que está muy en boga lo de AlphaFold, otra parte de los proyectos que se están haciendo en la empresa es justamente ver si los resultados que se obtienen con modelos de inteligencia artificial y no con estructuras experimentales son igual de buenos, y los resultados son bastante alentadores.
También comenta el doctor Rodrigo Aguayo: excelente trabajo y muy interesante, doctor Norberto. Le mando un fuerte abrazo.
Muchas gracias.
Y bueno, creo que vamos a tener tiempo para hacer un break. Le cedo la palabra al doctor José Luis.
Muy bien. Muchas gracias, maestra Fernanda, por dirigir esta primer bloque del segundo día del coloquio. Quiero agradecer a nombre de todos los organizadores, a todos los ponentes por sus excelentes presentaciones. Muchas gracias al doctor Miquel, al doctor Norberto. Muchas gracias a todos ustedes por sus excelentes presentaciones, desde luego también al doctor Eli. Pues vamos a tener ahora un receso de 20 minutos. Entonces, mientras tanto, si quieren llenar el cuestionario de asistencia que se va a poner en el chat y que ya también está en las redes sociales. Entonces, continuamos a las 11:50. Muchas gracias.
Muchas gracias a ustedes.
buenos días o buenas tardes, noches. Bienvenidos a la segunda parte del segundo día del coloquio. So, good afternoon. We're going to resume with the second part of the second day of the colloquium. And now I'm going to hand over the microphone to Master in Science, Ana Luisa Chavez. She's going to present the next two speakers. Muchas gracias.
Buenas tardes a todos. Gracias, doctor Medina, por cederme la palabra. Hi, everybody. I have the honor to present the next speaker, Doctora Raquel Rodríguez Pérez. Doctora Raquel Rodríguez Pérez is a principal scientist at Novartis Institute for Biomedical Research and works in modeling and simulation data science team in the Translational Medicine Department. She develops machine learning models to predict compound properties relevant in pharmacokinetics. She supports drug discovery teams with modeling and data science tools in order to make better and fast decisions in lead optimization. Prior to working at Novartis, Raquel obtains her bachelor and master degrees in biomedical engineering from the University of Barcelona and her PhD in computational life science from the University of Bonn. She worked on data analysis for bioinformatics application at the Institute for Bioengineering of Catalonia, and did her thesis about machine learning models for interpretable compounds activity predictions. Therefore, she has experience with the application of machine learning and deep learning methods in different life system science problems. She was a Marie Curie Fellow and early stage research at the Computational Chemistry Data Science Group in Boehringer, Germany. She has acted mentor of scientists and different careers develops both in academia and industry. Overall, her research interests include bio and chemoinformatics, machine learning, and data science for a biomedical application. Dr. Raquel Rodríguez Pérez, thank you very much for your presence. You have the floor. Muchas gracias por su presencia, doctora Raquel Rodríguez. Usted tiene la palabra. Doctora Raquel.
Dr. Raquel, I think you can share now your screen. You have the permissions now. I think she will be projecting very soon.
Okay. Doctora Raquel, ¿nos escucha?
Okay.
¿Usted puede compartir su pantalla?
Hola. Hi, everybody.
Hola.
Sorry, I was not listening to you until now. I had a problem with the speakers. So I start sharing my screen?
Yes, please. Go ahead with your screen. Thank you.
I'm sorry. Confusion. Perfect. Can you see it?
Yes, we can see your presentation in full screen. Thank you.
Perfect. Hi, everybody. And thanks for the invitation to present in this colloquium. Today, I will explain you first a brief introduction of who I am, where I work, and what we do, how we interact with other teams at Novartis. Then I will present some fundamentals of molecular machine learning. I will show you some key applications of molecular machine learning and data science at NIBR and with applications in the chemoinformatics field. And then I will show you a case example in which we develop machine learning models to predict whether a compound will penetrate into the brain. So who I am, I'm Raquel Rodriguez. I work in the modeling and simulation data science team at Novartis. And I would like to tell you a little bit about my background because I guess that here there are a lot of students and I was always very interested in knowing what people studied and what background they had prior to joining industry. And how does the work change when you move from academia to industry? So what I do currently in industry is developing machine learning models and apply data science tool to support decision making. And in particular, I work in the lead optimization stage. I studied biomedical engineering in the University of Barcelona, where I'm from. And then I moved to Germany with Professor Bajorat to do my PhD in computational life science. During that time, I also have some industry experience because I was doing an industrial PhD and I work at Boehringer Ingelheim in Germany. My research background is predictive modeling in bioinformatics and chemoinformatics. So I analyze different types of data sets. I started with metabolomics and biomarker discovery in a research group in Barcelona. We also work with chemical gas sensor arrays to detect different types of gases with an array of sensors. And then during my PhD, I focused on compound biological activity predictions to know whether a compound will interact with a target and produce a function or not. And I like getting insights from complex life science data using multivariate statistics and machine learning. And the focus of my work is help accelerating drug discovery. In the modeling and simulation data science team, we work closely with other teams, and this implies having a multidisciplinary background or working in a multidisciplinary environment. So you need to know a little bit of computer science, software engineering, and mathematics to be able to communicate properly with, for instance, the research informatics team. Then you also have collaboration with other data science teams that mainly focus on a different data set or a different problem, but we use very similar tools. And you also need to know a little bit of chemistry, pharmacology to closely interact with discovery teams, which are using your tools and your models, and also with PK scientists in my case, because we are part of the pharmacokinetics department, and I'm analyzing the data that they generate. So sometimes I have to go to the lab to know how the assays are working and how we can improve certain aspects of data science. And as I said, with all these close collaborations in a multidisciplinary environment, we aim at accelerating and improving decision-making in drug discovery. So machine learning is applied at different stages of drug discovery. I will first give you some exemplary applications, and then I will focus on molecular machine learning. So in the target identification stage, it's important to apply data science and machine learning to predict, for instance, suitability of targets for drug discovery. This can help accelerating this first stage. During the hit discovery, an important application is virtual screening or computational compound screening, in which you have large libraries and you can analyze the previous high throughput screening experiments to identify which compounds are promising for further analysis. And there are other applications also. I will focus later on lead optimization, but in preclinical development and clinical development, you can also help with machine learning, for instance, discovering cell-specific biomarkers, analyzing gene signatures. This is more in the context of personalized medicine to identify patient subgroups. Or another important application that we are also working on at Novartis is pathology image processing. So about compound property predictions, which is the main focus of this talk. In machine learning, we can apply different algorithms to make predictions about properties of the compounds. When we want to decide which compound is our drug candidate, we require multiple experiments to characterize the properties. But if we build this kind of algorithms that relate the compound structure to a given property, this will enable faster and more informed decisions, prioritization of compounds, scaffolds, or specific chemical modifications even prior to synthesis, and also an experimental selection. And here you see the general workflow. I will later focus on these different building blocks, but the idea behind these compound property predictions based on machine learning is that you can encode a compound using some molecular features, and then using this numerical representation to apply a machine learning algorithm that it's giving prediction for new compounds, for instance, high potency, or the application that I will show later, the brain penetration. If we focus on the molecular features, it's important to compare how the different encodings of compound structures numerically can influence the predictive performance. The standard descriptors are either based in 1D, 2D, or 3D representations, and another one that is very common are the molecular fingerprints. The binary molecular fingerprints encode the absence or the presence of certain chemical substructures or atom environments. So the absence will be with a 0, the presence will be with a 1. So for each molecule, you will have a binary vector that it's giving you the coordinates of this molecule in the chemical space that you have defined. For instance, in this three-dimensional representation, if you have a 0 in this coordinate, a 1 in this coordinate, and a 1 in this other axis, then you will project, for instance, this compound as a point in this space. But normally we are working in a higher dimensional space, and we will try to find patterns in this space. And the first step of supervised machine learning is to train a model. So you will require, for instance, a set of molecules with a low risk and another one with high risk. This could be, for instance, high potency, low potency. This could be low risk of penetrating the brain, high risk of penetrating the brain. And the model will learn structural patterns from this compound data. And for instance, it will find a hyperplane that correctly classifies these two classes. The second step will be making predictions for new compounds. So in this case, if the model is made to make activity predictions and you have two compounds for which you don't know if they are active or inactive, you could project these structures into this chemical space and then see in which side of the hyperplane they fall. And then you could make the predictions about these new compounds. So in general, molecular machine learning involves different steps. These are the classical ones of all machine learning models. It ranges from data curation to model evaluation, starting by preparing the data, extracting it from databases, doing some preprocessing of the chemical structures, analyzing the data variability. Then you start building some models. You select which one is the optimum and which one you want to estimate in its performance in an external validation set. Then you check how this model performs, and if that's successful, you can use it to support decision making. The main difference in this type of models is that we will use them in pharmaceutical research, so we are aiming at making the best decisions to bring medicines to patients faster. And now I will show you a key application, the machine learning for predictive safety. This is one of the first models that I developed when I joined Novartis, and this was for bile salt export PAM inhibition. So this is related to DILI, the drug-induced liver injury. This is one of the main mechanisms that leads to DILI. And DILI, it's a main cause of drugs failing in development or getting withdrawn from the market. So we want to have an early safety flag if we are developing new chemical series or compounds that might have this risk of DILI. We want to know early on. So we developed a machine learning model for this predictive safety endpoint, and we were aiming at the early identification of BSEP inhibitors. In particular, there is an assay which measures the IC50, and we, due to previous analysis, we knew that the IC50 lower or equal to 30 micromolar was considered a safety flag. So we developed the model based on machine learning methods, and the prospective evaluation indicated that the model was having a 90% precision and only an error of 4%. So when the model predicted that a compound had a safety flag and had the IC50 of BSEP lower than 30 micromolar, it was correct 90% of the times. So now the project teams have access to this model, and if they want to get rid from this BSEP liability, then they can check the in silico profile prior even to synthesis of the compounds. Apart from machine learning models, we also develop different interactive dashboards for drug discovery. This improves the data curation, instant access to intuitive visualizations, which are key for decision making. So if we have a protocol for data curation and we have some framework for visualizing model performance, correlation, toxicology, putting together a lot of virtual, in vitro, and in vivo data together for the project, then we can provide this tool to project teams. And the motivation of developing this tool was to have project-centric visualizations of all discovery data. So these kind of dashboards are very easy to use, even for non-data scientists. You have very complex visualizations that are automatically done for your project. And this, again, was aiming at accelerating drug discovery because it's easier to make decisions when you have these informative visualizations that imply curated data and are actively they are updated. Apart from this, we are also involved in state-of-the-art research in molecular machine learning. So a part of applying standard methods from machine learning or data science, we also see some challenges, and we want to explore how newer machine learning and deep learning models can offer opportunities and help with difficult problems in chemoinformatics. So novel methodologies are regularly developed or applied in-house for challenging problems. And for this, we have collaborations both with academia, but also other companies, for instance, with Microsoft, and we further investigate in these areas. And I will now show you 4 examples. The first one is generative modeling. So instead of having models to predict properties for compounds that have been designed by medicinal chemists, we could also train a generative model to optimize in this latent space certain scoring functions, according to a certain scoring functions or multi-parameter optimization. And in this latent space, we have property prediction models. So we are finding which is the optimum or which are different optimums in this space, and then we can obtain to the extractors that they are encoding. Another interesting approach is active learning. So instead of developing a model and have it static and then use it to make predictions, you can also use it to select experiments. And then for the new experiments that you've done, you can update the model and select it to do the next round of experiments. And what it allows is to achieve better performance with less data points. There are also some consortiums for the federated learning approach. For instance, Novartis is involved in the MELODY project. And the idea behind the federated learning is that different institutions can use their data to make local model updates. And then they have a global model, which is using data from different companies, but they are not sharing the data. And finally, an important topic is also model interpretation or explainable machine learning. So we would like to have intuitive visualizations to help medicinal chemists understand the decisions from the machine learning model. So why was this prediction made? What are the parts of the compound that are influencing this prediction? And now, after this overview of our work at Novartis and how molecular machine learning and data science can help in drug discovery programs, I will explain you a case example of prediction of compound brain penetration using machine learning. Brain penetration is the ability to reach the central nervous system and reside there a certain time. The blood-brain barrier is involved in the diffusion of molecules from blood to brain, and is controlled by several mechanisms. So for instance, some compounds can diffuse towards the BBB, but you can also have some active efflux. So MDR1, that it's also called PGP, glycoprotein 1, are an important mechanism. And they are also active transport, and there are also other mechanisms involved. Knowing if a compound will penetrate the brain or not, it's very important, and it's not only important for CNS drugs. So on the one hand, if we want to treat neurological disorders, we would like to achieve brain penetration to reach the target. But sometimes there are also compounds that are targeting for other targets that are in the CNS, but we want to avoid CNS-related adverse events. So one way of doing this is avoiding that they interact with the proteins in the brain. How can we measure brain penetration in the lab? There are different assays that are done at different stages of the drug discovery and development process. The in vitro assays are passive diffusion, and an important one is active efflux. Passive diffusion, it's measured through artificial membranes or cell layers. And the active efflux of a compound out of a cell layer overexpressing transporter is the primary assay to screen for compounds if you want to achieve or avoid brain penetration. In the advanced development, we also have in vivo assays where you measure the brain-to-blood concentration in preclinical species, normally rodents. How can machine learning help in this? A machine learning model can be developed for each of these assays. So for instance, in passive diffusion, we have the low efflux MDCK assay and the PAMPA assay, which will be the cell layer and the artificial membrane. And we have models for that. But even more important for this brain penetration assessment, we would like to have an MDR1 model and the KP model because are the main assays that are used to drive decisions about brain penetration. And the aim of this project was developing MDR1 and KP models to help in the design of brain penetrant or non-penetrant compounds. And for each of the models, the following steps were carried out. The ones that I presented: data preparation, model building, performance estimation, and finally using them for decision making. For both models, the machine learning setup was a time split. This means that we are training the model with all their data and try to predict prospectively the most recent experiments. The setup of this evaluation is important because we want to mimic a realistic application of the models. Now I will explain the MDR1 model development. The MDCK-MDR1 assay measures the efflux out of MDCK cells that are overexpressing the PGP protein. This informs, among others, of the BBB permeability. And as you can see in this plot, the efflux ratio from the MDCK-MDR1 assay, it's correlating with the brain-to-blood concentration ratio. The efflux ratio is determined by assessing the transport across the cell monolayer in both directions, apical to basolateral and basolateral to apical. And as I said, this is a primary screening filter for brain penetration because if the drugs have a high efflux, typically the threshold is considered at 6, this will have a detrimental effect on brain penetration. Typically, to account for experimental variability, it's recommended to consider a low efflux if it's lower or equal to 2, and a high efflux if the efflux ratio is higher than 6. and in between it's the medium class. For model building, we tested different machine learning algorithms and molecular representations, including, among others, super vector machines or random forests. The final model consists of an extreme gradient boosting regression, which was built with a combination of Morgan fingerprints and two-dimensional RDKit descriptors. RDKit is an open source library. The performance is reported here for the top performing model, which is the XGBoost, and you can observe in the x-axis the predicted log efflux ratio, and in the y-axis the experimental log efflux ratio. This is the performance on the calibration set, which we use for model selection. As you can see, there is a correlation between predicted and experimental. In particular, the correlation was 0.7, and the mean absolute error was 0.36, which is a lot smaller than the baseline of consistently predicting the average. The percentage of predictions that were with an error lower than twofold was 54%, which can be considered a successful prediction. However, if we want to use this model for decision making, it can be quite complex to know when 2 points are distinct from each other, because we have also some predictions with a high associated error, for instance, these ones here in orange. So what we did is to use this regression model, which provides a good ranking of compounds of 0.7 correlation, to build a classifier. And this was performing better than directly carrying out a classification model. So these regression-based predictions were binned into high, medium, and low, and the medium predictions were totally discarded. They were set to inconclusive or unreliable. And by doing this, we can achieve a much higher precision and a much lower classification error. When the model predicts that a compound has a high efflux, we want to be correct most of the times, and we don't want compounds switching from a high to a low category. And here you can observe how we binned the predictions from the regression model to obtain a classification on the validation set. So this was a prospective validation with approximately 2,000 compounds. The average error was 2.4-fold. 68% of the predictions were within twofold, and the predicted values were binned into lower than 2 and higher than 6 in red and blue respectively. All the predictions in gray were discarded. So more or less, we discarded 50% of the predictions, and 23% of them were correct and had a medium efflux ratio value. But for the practical application of the model, we don't mind that we don't have predictions for 50% of the data, but what we want is that when teams have access to a prediction, it's very likely to be correct. So if you observe the predictions from the model, when it predicted that a compound had a low efflux, it was only wrong 5% of the times. And when it predicted that a compound had a high efflux ratio, it was only low in 8% of the time. And now I will show you how we developed the KP model, which was a work from the TM Data Science Academy fellow, Seth Hamsik. The brain penetration assay measures the KP, which is the concentration in brain divided by the one in blood. And we typically used the values at 1 hour time point. Many data curation and filtering steps were applied because it's a complex in vivo endpoint, and to achieve and to have a large data set, we had to standardize many variables because the protocol has been changing in Novartis over time. So for instance, we considered rodent data, the route was IV and oral, we considered a lower dose, and the time points were typically 1 hour, but we tried to avoid the very early time points and very late time points. Then we defined the classes of brain penetrant and non-penetrant, which was a 10% threshold. So in log KP, it will be a -1. And as you can see, this data set was very imbalanced. So we had only 15% of the compounds that were not penetrating the brain. And for this class, I will call them VBB minus. After data curation, we had more or less 2,200 compounds. So as you can see, this data set was much smaller than the MDR1 data set. In this case, the standard machine learning models were not as successful as with the MDR1 model. And we had a small data set, and we were not able to achieve good performance for the minority class of non-penetrant compounds. So we started to explore more novel or state-of-the-art methods, such as multitask learning and graph neural networks. Multitask learning consists of modeling multiple tasks or essays simultaneously. So instead of building one model per each essay, you could build a model considering multiple essays. And this has shown to be effective for related tasks, and especially for endpoints with a small dataset size, which is the case for brain penetration. So what we did is to use in vitro assay data as auxiliary tasks with the aim of improving the brain penetration predictions. And the model that we used was a graph neural network where molecules can be represented as graphs. The atoms are the nodes and the bonds are the edges. So with this, you can learn the representation instead of having a pre-computed representation like a molecular fingerprint or the descriptors, which are the ones that I presented earlier and that are more standard in the field. To give you a little bit more detail about this multitask graph neural network, it was a directed message passing neural network based on the ChemProp library, and it was trained on multiple tasks simultaneously, which are listed in this table. All of them were time splitted, so we never had the same compound in the training and the test set. The related auxiliary tasks were about permeability, passive permeability. In particular, there were 4 essays. And then we had the famous MDR1 flux ratio as an additional auxiliary task. But we were focusing on the evaluation of in vivo brain penetration. In this case, we also consider a regression model as a classifier because it was performing better than directly training a classifier. This was an interesting finding also from this study. And once we had the predicted log KP value, if the value was quite high, then we were assigning with high confidence that the compound was penetrating the brain, so it's BBB+. And if the predicted log KP value was low, then we were assigning with high confidence a BB- on non-penetrant prediction. And the compounds in between, they were assigned to inconclusive or medium. And here I'm also showing the prospective evaluation, in this case for the KP model. The predicted log KP values that were quite high were predicted BBB+. So this is the red dots here, and the blue dots were the BBB-. As you can observe, the precision in the prediction of BBB+ was quite high, was 95%, but what it was more challenging was also to achieve a high precision in the minority class, which had only 15% of prevalence, but we were able to achieve almost 90% precision. And in this case, the number of inconclusive predictions was much smaller because we don't have a medium class according to the experimental values. And what I also want to show here, it's a comparison to more classical approaches. So typically, if we want to achieve brain penetration or avoid brain penetration, we have some physicochemical properties cutoffs that have been reported in medicinal chemistry literature. For instance, these 3 cutoffs based on topological polar surface area, or this rule-based cutoff and BBB score, which were published in JMed Chem some years ago, are based on very intuitive rules for medicinal chemists. But as you can observe here, the precision is only high in the majority class, but they are struggling to detect with high precision the non-brain penetrant. Moreover, Other standard machine learning models, such as k-nearest neighbors with 2 descriptors or random forests with 2 descriptors, were also struggling in this minority class. And only when we only achieved high precision in the BBB minus, so in the non-penetrant, when we were considering multitask graph neural networks, and when we were assigning between 25 and 30% of the predictions to inconclusive, then we were even achieving better Matthew correlation coefficient. So the machine learning models are less intuitive because we are using more features, more descriptors, but they also have more predictivity and they will allow better compound prioritization. And now I will give you a models overview, and then I will explain you how this is supporting decision making. So in summary, about the MDR1 model, we are modeling the efflux ratio from the MDCK MDR1 assay, which is a primary screening filter from brain penetration. If we have a high efflux ratio, in particular higher than 6 approximately, we will have a detrimental effect on brain penetration. The model was able to achieve very low misclassification rates. So when it predicted that a compound had a low efflux, only 5% of the predictions were wrong. And when it predicted that a compound had a high efflux, only 8% of the predictions were wrong. And this was a prospective validation with the most recent experiments. On the other hand, we also built a model for an in vivo endpoint, which is KP, the concentration in brain divided by the concentration in blood for rodent data. And we use a 10% to divide the brain penetrant and non-penetrant compounds, which gave a very imbalanced distribution. However, we were able to achieve high precision both for the non-penetrant with 90% and the penetrant with 95%. With 28% of inconclusive predictions. So now I'll show you an exemplary experimental selection based on machine learning models. So for a specific project, we predicted the KP and the efflux ratio for all the compounds that they were considering to make. And then we selected the new experiments according to 2 criteria. First, medchem interest for the project, because sometimes these are not the only 2 properties or the only 2 predictions that we want to consider, but maybe they have other predictions about activity, or maybe the compounds are not available. And then they selected, for instance, this compound, which was validated experimentally and the predictions were correct. Sometimes we need a successful project-specific evaluation because this increases the model acceptability and the confidence in the predictions. And here I have listed some user feedback. For instance, chemical modifications that seem very similar to chemists sometimes lead to different predicted values. So they are using this for prioritization. Some teams have also found these models very useful to score entire series. Sometimes they use it as a guidance and then monitor the performance of their series, and then they start to get confidence and use it as a stringent filter. So some ideas that they have, if they already see that, for instance, the compound penetrates into the brain and they don't want to have this safety problem, then just discard the idea. And then they are using the model to design new compounds. Apart from the experiment selection, prioritization of compound ideas or chemical series, and the guidance of structural modifications, we also have a couple of applications that maybe are not so obvious, which will be library design. And a very important one now is generative chemistry. So we can use these models to also generate compounds with desired properties, and in this case, try to achieve or avoid brain penetration. So in conclusion, apart of showing how molecular machine learning and data science tools are used in modeling and simulation and in general in NIBR, I also show you a specific example in which we develop MDR1 and KP models to assist in compound prioritization and optimization to achieve or prevent brain penetration. In this case, machine learning and in a multitask architecture enable the use of in vitro assay data to improve predictive performance on in vivo brain penetration. So the in vivo endpoint had low amounts of data, and we were able to achieve top performance with the multitask graph neural networks. these improved classical medicinal chemistry approaches and also more standard machine learning strategies. Importantly, the model performance was estimated prospectively using the most recent experiments, and this is important if we want to assist in decision-making with these model predictions. Uncertainty was also integrated in the model predictions because some predictions were flagged as unreliable or inconclusive, and this enabled to achieve a higher precision and lower misclassification error when we were actually providing a prediction to the teams. And sometimes to further increase confidence and model acceptability, we also need to perform some place-specific evaluation, which sometimes is challenging, especially if we are working on in vivo endpoint. So some take-home messages: data science and machine learning can support decision-making and accelerate drug discovery. Novel molecular machine learning methodologies are developed and evaluated in order to solve challenging problems. So here I've shown how multitask graph neural networks were developed in-house when we found a very challenge to model endpoint. But understanding the data and the property to model is essential. We need to communicate closely with the SAE owners and with the model users. So we need to know how is the data generation process, how the data is used in the context of drug discovery, and how the model will be used. And finally, data scientists in pharmaceutical industry work in a multidisciplinary environment. So we need domain expertise and high collaboration with our colleagues from other disciplines. So these are some of the references. These are works that we did related to the topics that I presented today. And I would like to also acknowledge to some colleagues that worked in this project and developed some of the models that I presented today. And of course, all of you for your attention.
Thank you very much, Dr. Raquel, for your excellent presentation. And we have some questions. Rafael from YouTube asks, to improve the MDR1 model, would you collect more data or try to find better descriptors?
Mm-hmm. Yeah, that's a good question. I think that in this case, the learning curve, so when you build a model with a different data set size and you monitor the performance, you can observe whether you have already enough data that saturates the performance. So for instance, for the MDR1 model, adding additional data was not improving the performance. Of course, if you are expanding to new chemical series and then you update the model, this would help. But if you are updating the model today with the data, with more data, then anyway, if the chemical space is shifting, you will see the drop in performance. So, and another answer related to this, it's that the multitask GNN also has an auxiliary task, the MDR1. So we are currently evaluating whether this is also improving the performance because it's using different descriptors.
Thank you.
Tu micrófono, Ana. Prender tu micrófono, Ana.
¿Me escuchan ya?
Sí, muy bien.
Sorry. The question was very interesting. I would like to know what kind of experimental activities are considered in the collaboration with the academia, and how close far in the link between academia and the pharmaceutical industry?
Mm-hmm. So with the experimental activities, so I I think when we collaborate with academia, we mostly use public data unless someone wants to apply an algorithm internally, but we don't share the data. So if that's the case, then we will need to apply the algorithm ourselves and then just share the results. I don't know if this is answering a little bit the first question. And most of the research in cheminformatics done with potency data or activity data, but now the ATME properties are also starting to be hot topic because we have more data in the public domain. So it's also easier to develop models. And about the link between academia and industry, I thought it will be a bigger change, but actually it depends a lot where you are in what are your tasks in your specific team. But we are working on similar problems. We are interested in similar problems. Sometimes in industry, we just try to find a quick solution because the teams need to have the solutions because they have deadlines and they want to have the predictions earlier. So we need to find a trade-off between how much we do investigation in one topic and how long does it take to provide the model. But in general, I think that we are interested in very similar questions.
Thank you so much. I have another question from YouTube, Luis Islas. Excellent presentation. I would like to ask you what kind of computer you need in order to use the tools you show us, or supercomputer, or just a very good personal computer?
Mm-hmm. Yeah, so it depends on the method, and the dataset size. So for instance, for the MDR1 model, which was around 10,000 trained data points, and it was an XGBoost. I don't think you need a lot of computational requirements. That probably you can do it with a good personal laptop. But for the multitask graph neural network that we developed, which I forgot to mention it, which is an ensemble of 5 multitask neural networks, and are also including a lot of data from in vitro assay. This one we use the HPC and in particular GPUs.
Thank you. Another question from Dr. José Luis Medina Franco. Excellent talk, Dr. Raquel. How do you compare the ATME models developed at Novartis with the models free available in websites used in academia?
Yeah, that's a good question. So we sometimes evaluate models that are already out there, either for commercial software or from academic groups. It depends on the composition of the training set from these models, because we have found that for some of the models it's quite it's quite good to use the ones that are already available, and you don't need to involve like internal efforts to develop them. But some of them are very different chemical space, so they are not really working for some of the projects. So what we are trying to do is to have a workflow to monitor the performance of these models that are freely available or that are in commercial software. So that we know for specific projects whether we can use them or not in a more systematic way.
Thank you so much, Dr. Raquel. Thank you so much for your excellent presentation. And we have 2 next speakers.
Thank you very much.
I have the honor to present. Thank you very much. I have the honor to present the new next speaker, Dr. Jordi Mestre. Good afternoon, Dr. Jordi Mestres. Dr. Jordi Mestres holds a PhD in University of Girona. After a postdoctoral stay at pharmaceuticals and a job in Kalamazoo, Michigan, USA. In 1997, he joined the Molecular Design and Informatics Department an organo in Oost, in the Netherlands, and 2000 he has appointment head of computational medicinal chemistry at Organo Laboratories in Newhouse. In 2003, he took his current position as head of a research group of system pharmacology within the research program on biomedical informatics at the Hospital Omar Medical Institute Research Institute in Barcelona. He also associate professor at the University Pompeu Fabra, and in 2006 he founded Chemotargets as an spin-off company of his academic group. He's also the recipient of the 2006 Berkeley Hash Award from the QSR Modeling Society and the 2000 Technology Transfer Award from UPF. In 2018, he was admitted as a fellow of the Royal Society of Chemistry. His experience and current research interests focus on the development of novel computational approach and the side of new analytic tools to predict and visualize the pharmacology and safety profiles of small molecule pharmaceuticals. He is the author of over 160 publications and 10 patents among them. Dr. Jordi Mestre, thank you very much for your presence. You have the floor. Gracias por su presencia. Le cedemos la palabra.
Thank you very much, and thank you for the kind invitation. I hope you can hear me well. It's a pleasure giving this last talk of the day. I know it's morning on your side and it's late. You must be very hungry. So I'll try to be brief and go to the point. The focus of the presentation is on data. Before attempting to construct and build any model, it is always good to pay attention to data, and especially to pay attention to how this data is labeled, structured, and you see that throughout the presentation. I took one picture from the internet. I guess you are familiar with it. And the main message here is that there is no learning without labeled data. If you ask to an audience who wants to do machine learning and apply scikit-learn, keras, and TensorFlow to a bunch of data available, I'm pretty sure that everyone in the audience would raise the hand. Machine learning, artificial intelligence is a hot topic these days, and everybody wants to be on top of it. Now, if you ask the question, who wants to provide data, and especially to provide data, not to generate data, and the data providers are not many today. So you can have data in the public domain, you have data in public repositories. For example, you have in vitro affinity data in Campbell, or you have spontaneous reports publicly available from the FDA adverse event reporting system. You have also commercial providers, for example, the good old Wombat affinity data, or the BGBase, which will be the counterpart of FHIR's data, but from the World Health Organization. And then you can also provide data from internal initiatives. And we do that. We do that a lot. We have a data extraction team that mostly focuses on preclinical and clinical data that might be harder to find than in vitro affinity or post-marketing data. And if you change the question and ask who wants to label data, then not even a single person would raise the hand, but they would disappear completely. Data labeling and structuring data is not an easy task, is not a very rewarding task is not highly motivating, but it's extremely important, as you will see in the next slide. So we put a lot of effort in annotation and curation, because we have observed that well-curated and annotated data brings real added value when we then attempt to build models. But of course, it's extremely time-consuming, and there are parts that can be automated, but other parts can simply not be automated, and they have to be done manually and curated visibly. So that's the situation we are facing, and we're putting a lot of effort in data analysis, labeling data, annotating data prior to attempting to construct any model. Now, here the paradigm for the previous years has been from target drug discovery, target-directed drug discovery, to the systems of discovery. We used to work in a world where we would be having in vitro affinity data that would connect small molecules with protein targets. And now we live in a world where data is collected from all different angles of a biological system. ATME, we've seen some models for ATME properties, metabolites need to be predicted. What are the organs where the targets are expressed? In which biological pathways these targets are involved? What are the side effects which are associated with having affinity for those targets? What are the targets that are relevant for certain diseases? But also, what are the personal properties or circumstances that might make that a drug molecule works for certain individuals and it doesn't work or is not as efficient for other individuals? So all this is now providing a wonderful opportunity to develop a whole new generation of platforms that integrate all these data into models to anticipate side effects. And we normally talk about side effects and safety data, but across the lifetime of a drug, there is safety relevant data being collected at different stages. And I've tried to summarize when and where safety data is being collected across the lifetime of a drug. Normally in pharma industry and even in academia, a drug discovery project starts by screening a large chemical library. And the output normally is a handful of small molecule hits. And once we confirm the structure of those hits, we try to derive a dose response curve. Now, these in vitro affinity data, it's already an early relevant safety data for the drug. We can now connect some of the in vitro affinities for certain proteins to side effects that might occur later on in the lifetime of a drug. Now, some of these small molecule hits will progress into heat optimization and eventually become a lead. And then new safety relevant data will be acquired, first in vitro assays. Let me see if I can... In vitro assays that give you some preclinical data, but also in vivo safety data on preclinical animal models that give you some preclinical data as well. And then eventually, very few of those small molecules that you screen here will go into clinical trials. And you will get safety data here as well, which might be very different from the preclinical endpoints that you tested in the previous stage. And if everything goes well, that drug will go into the market and will be exposed to millions and millions of patients and healthy people sometimes that will take that drug. And you will get also safety relevant information from spontaneous reports from many reporters. Those reports can be from the patient itself, they can be from the doctor, they can be from the pharmacies, they can be even from lawyers that are faced with situations that patients sue a pharmaceutical company because they took some drug for a certain thing and suddenly they suffered non-wanted side effect. So all this data is collected somewhere across the lifetime of a drug. And we face a situation that no one until now that we know has put effort in actually connecting this translational safety data. So we can learn from in vitro affinity data all the way to preclinical endpoints, clinical safety, and post-marketing reports. And this is what we've tried to do in the last, I would say, 18 months that resulted in the development of a translational safety platform that allows you to integrate safety pharmacology data, preclinical talks, clinical safety, and post-marketing reports on a single platform and connect all this data to extract and identify signals once you process those well-structured and connected data into models. You have to face different challenges that we'll see in the next few slides. You have different data types, you have conflicting assessments, and sometimes you have data biases and completeness issues that you will have to somehow be aware of. But before we get into that, it's very important to know the volume of the data we are considering. And it's very important to understand that when we're talking about safety, it's just a tiny portion of the chemical space. Normally, when we talk about chemical space, now we're talking about the 4 billion or 20 billion small molecules that might be available in a virtual library that is often called real, which are not real, are virtual. But the number of small molecules, or the number of products, not only small molecules, therapeutic products for which we have safety data is limited to around 12,000. 9,000 single drugs, 12,000 drug combinations. And we can have antibodies, we can have biologicals, we can have peptides, we can have combinations, etc. So it's very important to understand that what we're talking about here is a tiny portion of the chemical space. And this tiny portion is organized in therapeutic areas. You can use the ATC, the anatomical therapeutical chemical classification, and you can see that most of the drugs that we can assign an approval year, come from the anti-neoplastic and immunomodulating agent. We have 331. Now, if we go into safety pharmacology in vitro affinity data, we have affinity data that covers about 3,800 targets, and we have roughly 390,000 in vitro affinities. And as you can see, a good portion of the in vitro affinity data is associated with protein kinases. You see it here in the light blue. We have also a lot of data on GPCRs, for example, and also quite a few data for enzymes, for other enzymes other than kinases. If you're talking about data heterogeneity, it's very important to understand that the concept of in vitro affinity may be associated to actually different data. You can have KIs, you can have KDs, you can have EC50s, you can have IC50s. So it's very important to be able to go deep down into the raw data, and sometimes you can explore constructing a model with all the data, an ensemble of data, regardless of the affinity type. But sometimes when you have enough data, it might be also interesting to develop models that are consistent on the data type. And you'll see that what I mean in a minute. So as you can see, there's a good portion of data which is KDs, but also we have a lot of IC50 data. In terms of preclinical data, we have about 84,000 preclinical data, and also we have about 5,000 drug metabolites, known drug metabolites, not predicted, just known. We're talking about known data, no predictions here. First of all, we are collecting known experimental data. And you can see here that a good portion relates to the digestive and excretory system, and also to the circulatory system. Now, going to the clinical safety data, we have about 88,000 safety data directly from the drug labels. You know, when you know this piece of paper that it's inside the box when you buy, when you go to the pharmacy and you buy a drug box. That piece of paper that normally you reject or you throw to the bin, that's the drug label, and there is a lot of information. There is a lot of safety information there. So we use text mining and then manually curating what we extract from the drug labels. And also we have a data extraction team to extract data from all the tables in the medical reviews, the original medical reviews of every single drug. So you can see here that a good portion of the clinical safety data is associated with the nervous system. All safety events are linked to a nervous system, but also we have a good size of data for the circulatory system, or again for the digestive and excretory system. And finally, the post-market reporting data. We have about 50 million reports currently. About 20 million come from a public repository, which is FIRES. This is the FDA Adverse Event Reporting System. And we have about 30 million coming from BGBase, which is the database from the World Health Organization. We are working on integrating 2 additional databases that will be available in the next release of the platform. These are VIRES, which are vaccine products, and also JADR, which is the equivalent of FIRES, but for the Japanese market. So all these will be integrated, are currently being integrated, and will be available soon. Now, with respect to safety data heterogeneity, there are several aspects I'd like to highlight. So it's important to be aware of the different types of data, even though they reflect to the same concept, in vitro affinity or interaction, drug-protein interaction. We have different data types. And this is a very nice example of a given drug-target interaction for which we have data from all different types. So we have PEC50s, we have PIC50s, we have PKDs, we have PKIs. Sometimes we also collect the Campbells directly from Campbell, since they provide it, it's good to have. And we have also qualitative assessments, is active or inactive. And you can see here that depending on the type of data, the mean value is actually quite different. It can go from sub-nanomolar affinity if you take PEC50 data, or around 50 nanomolar if you take IC50 data, for example. So it's very important to to deconvolute all the data available into data types. Also, it's very important with respect to assessment. You can have conflicting data when you integrate data coming from different sources. So, for example, in this case, we have preclinical tox data associating rimonabant with white blood cell count decrease. And you can see that in one study was treatment related, in the other study was not treatment related. So you have to balance that, and you have to provide a certain score to reflect the fact that all assessments agree, or there is a disagreement or conflicting evidence with respect to associating a causal relationship between the drug and the safety event. Also, I'd like to stress here one of the points that a good data scientist should be aware of. What we read from the original report is leukocytes decreased. And what we translate into the database is white blood cell count decrease. So we have a huge number of what we call verbatims. Verbatims is everything that you read directly from the report. And we have millions and millions of them. Imagine that in the drug field, you can have the name of the active ingredient, pergolide, but you can also have the name of the product, Permax, or you can have the name of the drug and the dose, Permax 400 milligrams, and you can have Permax 200 without the space milligrams, so all together. So all these verbatims, you have to annotate them to a main drug name, which might be pergolide or might be Permax, but you need to recognize that all these verbatims are associated to the same concept. So this is a very important aspect of our data structure and data annotation activities. Now, with respect to clinical safety, it's very important to differentiate between an event and a reaction. An event is essentially any side effect that occurs to the patient while taking the drug, but there is no causal relationship. No causal relationship can be established between the intake of the drug and the side effect. Instead, a reaction means that there is a clear, or as clear as possible, causal relationship between the fact that the patient took the drug and a side effect was observed. And finally, the data that we face using post-marketing reports, you get data like that. You have dates, you have the route of the drug, you have the indications, sometimes indication, but others, some indications, multiple indications are annotated. You have the outcome, you have the age of the patient, you may have the sex, and you may have the country or the world region. So all this information is collected and somehow normalized and structured appropriately. So as you can see, there is quite an heterogeneity of data. And it's also very important to identify bias and completeness, as you will see in an example in a minute. Bias from, for example, from people reporting a certain drug or certain side effect, or completeness, like in this case, that some of the reports do not have complete all fields completed. Now, based on that, for example, for post-marketing data, we perform first what we call a safety assessment. We read a lot of data from this post-marketing reports, and every drug event pair is associated an adjective. So based on frequency, it's very rare, rare, uncommon, common, or very common. Also based on seriousness, it can be non-serious, it can be involved in serious. So the report contains a serious side effect, but there is no direct connection in the fields of the report connecting that particular drug with the side effect, or it is suspected serious. So in the report itself, there is an annotation that this particular drug is probably the primary suspect for this particular side effect. If we have this information, that is a suspected serious. And based on that information, we process it, and every side effect for every drug, it's annotated as an ADE, adverse drug event, which means that we cannot establish a causal relationship, or it's annotated as ADR, adverse drug reaction. So there are evidences that make us to suspect that there is a causal relationship between a particular drug and a side effect. Now, ADRs, we differentiate them in two different levels, depending on certain properties, certain criteria. We call them an ADR warning, and depending if those criteria are high enough, we call them an ADR signal. So is highly disproportionate compared to the reporting of that particular side effect for a reference set of drugs. Now, let me give you an example here. So if you go to the label of Erbitux, cetuximab, so I'm not talking particularly about a small molecule, in this case, it would be an antibody. So you go to the label and you see that there is a warning about dermatologic toxicity. So when we perform the analysis of cetuximab with respect to skin toxicity using post-marketing reporting data, we see that when we received the very first reports early in 2002, the skin toxicity side effect was labeled as an event. So no real causal relationship could be established. But then as more reports became available, we could progress this labeling of the safety event to an ADR warning. And finally, after 2010, the label achieved the level of ADR signal for the remaining of the years up until today. So we could detect skin toxicity already in 2011, a few years later than it was available in the market, which confirmed some of the side effects observed at the clinical stage. And that's very important. That's what you want to do in drug surveillance or pharmacovigilance. You have a long list of side effects that were observed at the clinical stage, and they may be or not causally related to the drug. And then what you do is you take this long list of side effects that are all written down in the drug label, and you check whether once the drug was exposed to millions of patients, that side effect becomes disproportionately related to the drug and it's a real ADR signal, or simply it's an event that is seldom reported. Now, this is an example of a reporting bias. So regardless of the database, if you do an analysis of all adverse events, you see that 46% of the reports come from consumers, and 21% of all reports deposited in files come from physicians. Now, if you go to side effects such as sleep disturbance, you see that this proportion is changed. So you have a bit more of consumers that report sleep disturbance than physicians. But on the contrary, side effects like thrombocytopenia, you see that it's the other way around. So much less consumers report thrombocytopenia than physicians. Now, if you go to a completely different database, such as BGBase, you see that the numbers are different, but the trends are similar. So the base numbers here are 16 and 33, but sleep disturbance is, you see an increase in consumer reporting when when observing sleep, when reporting a sleep disturbance, and you see an increase in physician reporting when reporting thrombocytopenia. So these are aspects that need to be considered when analyzing all these post-marketing database. And let me show you one very interesting example that we managed to identify, because normally we are interested, for example, in identifying safety events that might be affecting an entire drug class. So, for example, this is the class of PARP inhibitors. We have 4 drugs in the market currently: olaparib, rucaparib, niraparib, and talazoparib. And if you don't do this stratified analysis based on reporters, regions, ages, sex, etc., you might think that the distributions of the reportings are very much similar across all drugs. Now, if you carefully look at the origins of the reports coming from the different drugs, you suddenly realize that the reports for olaparib and talazoparib are mainly coming from European physicians, whereas the reports for rucaparib and niraparib are mainly coming from American consumers. So that is something you need to take into consideration when you actually do an analysis of these side effects for a particular class. Because then when you go in detail and analyze a sleep disturbance, you recover exactly the trends that we saw in the previous slide. So regardless of the fact that olaparib is being reported mainly by European physicians, when you look at a sleep disturbance, this side effect is mainly reported by American consumers. And the same for talazoparib. But when looking at thrombocytopenia, even though generally rucaparib and niraparib were being reported, the side effects were being reported by American consumers, the main reports for thrombocytopenia come from European physicians. So these are all fine details of the data that you need to consider when attempting to build models for that. And with respect to data completeness, that is also a good part of the efforts that we are doing. As I showed you and mentioned to you a few slides before, not all reports are complete. There is a lack of data, and we are using something that is called the BG grade, which is a completeness score and quantifies the amount of clinical relevant information in those reports. The completeness score ranges from practically 0 to 1, and normally values above 0.8 are considered well-documented reports. And the presence of clinical relevant information is assessed on these 10 categories. We have, for example, indication and dosage, which reflect something related to the drug. We have time to onset, so when the side effect appeared after the drug was taken, and the outcome, whether the patient was hospitalized or died, for example, which might be related to the side effects. We have also information related to the patient, so sex, age, country, which might reflect ethnicity, for example. And finally, we have some information of the report itself. So primary reporter, whether it was consumer or a physician, the type of report, and some additional comments. So based on that, you have all these categories, and you put a score depending on whether the information is available or not, and depending on the different adverse drug or side effects that are associated in the report to the drug, you end up with a completeness score for the different drug side effect pairs. And at the end of the day, the BG grade is just the average of the different completeness scores for the different side effects in that report. So that's also a good thing to check the data completeness in the data you are using. And finally, when you've digested all the data, you need to realize something that is very important. I mean, we are having data that comes from different stages of the drug discovery pipeline, pipeline, and we need to understand how we connect that data so we can learn on how this data is translated across the lifetime of a drug. So, for example, in safety pharmacology, we extract in vitro affinity data. Let's say we have identified that your lead compound or drug candidate is an agonist for serotonin 2B. Immediately, that is connected to cardiac valve disease. So you may raise an alert to that. But also, you may have collected preclinical toxicology data on an endpoint that is called heart rate disorder. You may have collected clinical safety evidence that is annotated as cardiac failure, and you may start having reports that associate a particular drug to aortic valve incompetence. So as you can see, you have different safety terms, but one thing they have in common is the organ. They are all associated with heart, and also the organ system. They are all linked to the circulatory organ system. So we can use the organ system to glue all the data in a seamless translational safety continuum that allows us to then start building models that identify connections between having affinity for serotonin 2B and observing aortic valve incompetence down the line at the post-marketing stage. Now, this is how we have implemented our system. So you have safety pharmacology data, and it goes here. You see the different colors reflect the different organ systems, so circulatory and integumentary, neoplasms, etc. So this is the barcode of organ systems at the safety pharmacology stage. This is the profile, the safety profile of this particular drug at the preclinical, based on preclinical toxicology data. This is the safety profile using clinical safety data for this drug. And this is what happens when you expose the drug to the entire population. As you can see, first, there is a lot of variation in the distribution of the different side effects observed for different organ systems, but eventually everything converges into something that is distributed across the organ systems, and after a while, it doesn't really change. So once you have a certain amount of reports, that is fixed into a given safety profile. So that's very important. This is when we can conclude the actual side effects, which can be causally, most likely causally associated to a particular drug. Now, just let me show you some examples. This is pioglitazone. So this is the evolution of the number of reports over the years. As you can see, it started with 50 reports, and now we have almost 70,000 reports. And you see how the importance of the different organs was evolving over the years. You see that suddenly this yellow region here increased dramatically. And actually, this is related to neoplasms and urinary, in particular, urinary side effects. And we could detect that bladder cancer was already an adverse drug reaction, so most likely causally related to pioglitazone already on 28th of August 2006. And this drug actually in 2011 was withdrawn in France and Germany due to the high risk of bladder cancer. So this is a nice example of how you can use systems like that to anticipate potential serious adverse events that might lead to a drug withdrawal later on. Again, going back to identifying drug class safety signals, this is what we collect. So, for example, for this class, we collect the different events for how many drugs in the class the side effect achieves the level of ADR signal, for how many drugs achieves the level of ADR warning, how many ADRs in general, ADs, etc. So we can identify the what we would call class signals. Now, when we're talking about class signals, we have first to decide what is a drug class. And it depends. I mean, we can have chemical classes, we can have target classes, we can have mode of action classes, we can have therapeutic classes. And I just selected one example of a chemical class and one example of a target class. So this is an example of a chemical class. This is the thiazolidinal iones, and these two drugs, pioglitazone, rosiglitazone, as you can see, they share this particular fragment, so they belong to the same chemical class. But surprise, when you look at the data, even though they look similar structurally, and several methods might struggle to really predict that similar chemical structures would have completely different safety profiles. When you look at the data, this is what you see. For pioglitazone, you see that the main region is linked to side effects associated to the renal and urinary system. For rosiglitazone, the main region of concern are side effects linked to the circulatory system, whereas for troglitazone, the main reason of concern is associated with side effects of the digestive and excretory system. And indeed, pioglitazone was linked to, heavily linked to bladder cancer, rosiglitazone to cardiotoxicity, and troglitazone to hepatotoxicity. Moving on quickly, let me show you one example of a target class. So you can have a chemical class within a target class. In this case, the example that we saw previously, olaparib and talazoparib, belong to what we could say the same target class, but this is in fact a target class. They are all PARP inhibitors, and as you can see, some of them have different structures. But when you look at these, what we call translational safety charts, you realize that they all have, as an ADR signal listed here, a side effect called anemia. So we are able to identify that anemia is a common ADR signal across all PARP inhibitors. And this is again how we immediately identify using all these well annotated data that anemia is a class event for the entire PARP drugs. Other examples, for example, ketoacidosis is a class signal for SGLT2 drugs, or for example, rhabdomyolysis is an adverse drug reaction that is common to all HMG-CoA reductase inhibitors. So very quickly, you can identify these class safety signals. And now the beauty of it, if you have well-connected and structured data, you can start identifying connections between the safety events at different stages, but also connections within the stages themselves. And I don't want to go into that. The focus of this talk is data, not in the models, but just to have a slide that we use all the structured data to finally end up with models that derive what we call signatures for different events. So, for example, we can derive a target signature for a side effect, or we can derive a side effect signature for a given target, or reaction, an ADR signal signature for a given ADR signal. So within post-marketing, and I collected some examples of it. So, for example, these are the type of target signatures that we link to safety. So, for example, to anemia, this is the ordered list of targets that are mostly associated to anemia, or to urinary tract infection, or to blood creatinine phosphokinase increase, or to Cushing syndrome, for example. So if we take the top target, that would be PARP1 in this case, SGLT2 in this case, we just disregard the cytochromes. All drugs need to be metabolized, so this sometimes happens that metabolite P450s get in the list. The HMG-CoA reductase is in here as well, linked to blood creatinine phosphokinase increase, and the glucocorticoid receptor linked to Cushing syndrome. Now we can do the other way around and interrogate what is the safety signature of these targets. And as you can see, we can recover exactly the same safety events. So the main safety issues associated to these targets are anemia, urinary tract infection, blood creatinine phosphokinase increase, and Cushing syndrome. And you also have a list of side effects which are associated with this target. Now, a warning here. Now, the fact that all these side effects are in this signature associated with the target, it doesn't mean that they are causally related. They just mean that they may have comorbidities with anemia. But the interesting thing is that if you look at the organ systems that are most relevant for the signature, you see that the organ system signature is quite different from one target to another. So here you mainly see side effects associated to the muscular system. Here you see issues mainly associated to the circulatory system. So there are features essential that distinguish what are the side effects mostly associated to particular targets. And just to finish, I think this is also a very good example of how we use this nice, well-structured body of data to identify what we call prior safety markers. So as you can imagine, when you get the first 50 reports, or the first 100, or the first 500 reports at the post-marketing stage, you will see that there is a salient feature. Some side effects are reported more often than others. And this is translated in the fact that a signal for a side effect will be identified earlier than other side effects. And I like to put emphasis on the fact that for the statins, which is a very well-known case, you see that myalgia is always the very first ADR signal that is identified. Now, we realize that myalgia was always the first ADR signal identified. But then later on, in all of them, we also saw that rhabdomyolysis was identified. So can we use myalgia as a prior safety marker of rhabdomyolysis? Now, we think that myalgia is a prior safety marker and can be used as an alert that when myalgia is immediately observed in the first spontaneous report as an ADR signal, you have a high probability that rhabdomyolysis will emerge later on as a side effect of concern for that drug. And why is that? Well, I think we found a very decent explanation for that. Myalgia is a medical term for muscle pain. Everybody understands muscle pain. Everybody feels muscle pain. So consumers, mainly consumers, report myalgia. And they will be the first to report muscle pain if they take a statin. Whereas rhabdomyolysis, now the diagnosis is supported by a urine test and a blood test. A consumer will not have access to it. The results of the test will be first available to the physician. So as you can see, myalgia is mostly reported by consumers and first. These are the first reports that we get, and rhabdomyolysis is mostly reported by physicians, and therefore comes later in the signal detection. So just to give you the message that over and above of detecting prior safety signals, we are detecting what we call safety clouds. So some side effects that concurrently occur and are reported and identified as signals across the lifetime of a drug in the market. And we are now collecting all these data, all these safety clouds that appear across the safety lifetime of a drug. And just to finalize, a very busy slide, but I didn't want to finish without showing the fact that all this is being used to identify connections between some side effects of organ systems that have nothing to do. I mean, myalgia, in fact, has something to do. It's muscle pain, has something to do with rhabdomyolysis. But the interesting bits are side effects that are associated with a given organ system, and those could be an alert, a very early alert of something that has nothing to do with it and happens in another organ. So, for example, the strongest connection here is between the immune and lymphatic system, so side effects associated to the immune and lymphatic system, and the integumentary and exocrine system. You can see the strong bar here. But there are also very strong bars associated between the side effects of the circulatory system and the nervous system, for example. So we have now all these connections, and we are now exploring what exactly this means. This is still in the exploration phase. So just here to conclude, I think if we need to learn from safety data, we need good structured safety data first, and I hope that I've presented enough examples for a case. Good structured safety data is not easy to find and generate. It takes a lot of effort. And particularly in the field of translational safety research, the main challenge is data heterogeneity. We've seen data types, different assessments, different biases, different completeness. These are all aspects to take into considerations. The benefits of having well-structured safety data are visible when building models, not particularly machine learning, but models in general. And actually, this is a never-ending activity. You think that once you've cleaned and structured your data, you're done. No. Once you've done that, the new update is submitted to you, and you have to do all over again, because you may identify new verbatims that you will have to map to main names, etc. So it's a never-ending story. It's a full-time job. So thank you very much. I'd like to thank all the people that has been involved in this particular area of research, and finish with very nice pictures of Barcelona and our research park. It's not Mexico, but it's as close as it can get. Thank you very much for your attention.
Thank you very much for your excellent presentation, Dr. Jordi Mestres. I have some questions coming from the Dr. Tudor Oprea. Dr. Tudor Oprea, you can open your microphone to ask to the Dr. Jordi Mestres.
Hola, Jordi.
I mostly put comments as I saw the slides. I really enjoyed your talk, and it's a really good exposure to the Clarity system. Sorry, and some of them were just comments, and they're all in the chat, and mostly to give you an opportunity to discuss further about the intricacies and difficulties of annotating the data. So, for example, when you talk about 10,000 drugs on one of your slides, I try to count them as much as possible. I cannot get beyond 4,700 when I look at active ingredients. And also you say 3,700 targets on the human side, and mode of action, it's probably closer to 700. It's not, there are not as many targets as it looks like. So those are comments, and of course it's just a different way of counting, because you have to count all the targets that are annotated, and if there is rat and mouse targets, of course you have to count them, because sometimes they don't have a human equivalent, and that human equivalent might be missing because no one measured it, but if they did measure it, it would be there. So now an interesting question has to be about MedDRA. When I look at the FIRES and MedDRA, when you map the preclinical, I assume you map them to MedDRA, right?
That's right, yeah.
So would it be, does it have the preclinical tox enough resolution to tell you which type of white blood cell is decreased? Because leukocyte is literally covering at least 5 cell types.
Yeah. So that information is not there. I mean, you know the information that is available in FIRES.
Okay.
And sometimes it is difficult to actually map it to a MedDRA term. So that's why we have this verbatim list of text that we then need to curate and associate to a MedDRA term.
So do you offer services to pharma companies to go back to their lab notes for animal pathology to try to get this as accurate as possible? And no, I put an example here because you had just the next slide on your talk, you had about the severity of the adverse event. So for example, if say a neutrophil count is 6,000 and it's decreased by 20%, it is decreased, but it's not life-threatening. But the neutrophil count of 0, that's a medical emergency. You have to go to the hospital immediately because you can die in 24 hours.
Yeah, so no, we actually extract, we have some preclinical reports, but those, we don't extract information from the reports directly. We actually collected information from already the data that was examined from those reports. So for example, for preclinical talks that you are referring to, some of the data come from the eTalks project and the publicly available from that.
All right.
So it's not that we do text mining on the report of pharma companies. We could offer that, but no, we don't do that.
I think you should consider it. I happen to know that certain companies are annotating their internal reports.
Yeah, well, that's one of the things. So if you have annotated and structured data, you can upload the data into the system, and then all connections will be made, the models revealed, etc.
Okay.
Yeah.
And then I also put a note, I tried to figure out how would the sodium cotransporter 2 be related to UTI, and the best I can guess is that if you fail to uptake the glucose from primary urine, then it gets concentrated into the secondary urine, and that becomes a breeding ground for opportunistic bacteria that might enter by accident. Normally urine is sterile, but as soon as you give it a food source that's readily available, you know, it's dark, warm, glucose, and then bacteria just breeds. And I think that's really the relationship. And so it's a causal relationship, in my opinion.
Right. Yeah, I have to trust you on that. One of the beauties of doing this analysis is that I cannot interpret the data as you just put it. So we just identify relationships in an unbiased way. So the signal is there, and the good thing is that there are all the side effects associated to the urinary system that might also emphasize the signal.
It's I don't want to take over the sorry.
No, just perhaps I could comment on the number of drugs and products. So the 12,000 are products, not drugs. So they are products. They can be biologicals, antibodies, drug combinations. So we have a lot of drug combinations, and they are there because we identify them in the post-marketing reports. So we have these 12,000 products, and the 3,700 targets. I mean, they are actually proteins. And I don't know if you saw the percentage of drugs for which we have actually a mode of action target. I think this is about 13 or 15%. So you have to.
That sounds about right. That sounds about right. Yeah.
Very nice seeing you.
Likewise.
Thank you so much. Thank you so much, Dr. Jordi Mestre, Dr. Tudor Opera, for your question. Le voy a ceder la palabra al Dr. José Luis Medina Franco.
Okay. So thank you so much, Ana, for guiding this second part of the day. So I want to thank Jordi. Jordi, muchas gracias por unirte a la conferencia.
Ha sido un placer. Muchas gracias por la invitación.
Mil gracias. También Tudor, por unirte a todos los ponentes. Thank you so much for joining the second session of the colloquium. I want to thank all the speakers and look forward to seeing you tomorrow, 9 a.m. Mexico City time, for the third and last day of the colloquium. Thank you so much, and have a great afternoon, great evening, and see you tomorrow.
Thank you.
Bye-bye.
See you. Bye-bye.